All right. I think all the IT stuff seems to be in a row. We are now live on YouTube, and I'd like to welcome everyone. Good morning, and welcome back to the 6th Annual Plant Science Symposium at the University of Florida. For those of us who were unable to join us yesterday, my name is Josh Anderson, and I am the president of the Plant Science Council. Yesterday, some among you participated in our networking session, and other fellow students participated as student speakers. We have a fantastic lineup of invited speakers today. I'm very eager to hear from our Flash Talk competition as well. The competition winners for the Flash Talk will be announced later today at about 1:50 p.m. Eastern Time, right before our closing remarks. But before all of that, it's important to note that none of this would be possible were it not for the support of Corteva Agriscience. As mentioned yesterday, these symposia provide opportunities for the next generation of scientists to explore their interest in the plant sciences by inviting and hosting public and private experts on the topics of their choosing. And now I'm going to hand the floor over to Sudeep to introduce our first speaker.
Thank you, Josh. Hello and good morning, everyone. My name is Sudeep Kumar, and I'm the Vice President of Plant Science Council. Now I would like to introduce our first speaker. He is the Academic Relations Manager at Corteva Agriscience, Dr. Jason Rosser. He has been our advocate and has helped us through the symposium planning process, and he is here today to help kick off our Plant Science Symposium. Dr. Rosser is passionate about science and education with over 20 years in academia, research, secondary education, and industry. The perfect person to foster interactions between academia and the private sector. He currently manages the Plant Science Symposia series for Corteva, which supports a large network of graduate students, organized symposia at over 40 universities and research centers around the world each year. He also manages the New Frontiers in Applied Science Conference series and supports Corteva's intensive program for undergraduate and graduate students within R&D. So I'd like to introduce Dr. Jason Rosser from Corteva Agriscience. Dr. Jason Rosser.
Thank you so much, Sudeep and Josh. Can you hear me okay? Good, excellent. Well, thank you, everyone. It's a pleasure to be with you again here on the second day of Florida's Symposium. As Sudeep mentioned, my name is Jason Rosser. I'm the R&D Academic Relations Manager at Corteva Agriscience, and I'd like to just take a few minutes to talk about our program, and especially as this is our first event of the year, so give a little kind of wrap up of how last year went and what we're looking forward to this year. So slides will advance here. There we go. So I know most of you who are connected here today probably are familiar with our program. For those of you who are not, this is a Corteva-sponsored global program of scientific symposia. And the key here is that, as you will notice, this is all run by graduate students. And so this is our model for supporting graduate education and empowering the next generation of scientists, which we think is so important. And we do this in four different ways is kind of the way we look at it. In addition to the learning that goes on, it's a critical component of this program that students are given the power to choose the speakers, choose topics of their interest, and really kind of drive their education through these symposia. But the networking is a critical component as well. So, we had a really nice networking session last night, and the opportunities for students to meet speakers, interact with them, and build their networks as they're growing their careers and looking towards the future. We all know that networking is probably one of the most important things that they can be working on to make sure that they have all of those opportunities that they're looking forward to in the future. And speaking of those opportunities, we recognize that on the career side, sometimes in academia the private sector is a little bit of a mystery. And so we think these symposia are a great forum by which public and private sector scientists, professionals can come together with students and really have an open conversation and give students an awareness of what these different careers are really like. And so and that's really one of the most important things, and one of the reasons why we provide a Corteva speaker for each of these events. Dave Bubeck was our speaker yesterday, so I hope you had the chance to catch his talk. And then finally, what you'll see on display here are all those skills that these student leaders bring to the table. So the leadership, the teamwork, the management. These are kind of those things that aren't necessarily in a graduate school curriculum, but are really important for their future careers, whether that's private sector or public sector. And so this is really a great way for students to build some of those skills and show them off in this networked, high profile symposium. So those are our objectives there. We've been around since 2008. Kind of the history, started at the University of Minnesota, and then, it was so successful that it's spread quickly. And it's really, we've targeted the elite graduate programs around the world. And it's clear that Florida is one of those most important ones on our list. And we have some amazing connections. We continue to have amazing connections between our company and the university. Especially now with Charlie Messina, one of my former colleagues who's now a faculty member at University of Florida. I'm really excited about the future of that. But this is obviously an international network worldwide. We're growing when we can. We have really good roots in Brazil, Latin America. We're growing in Asia. We had some new events last year, for example, in Indonesia. And we're exploring some others in Asia as well for the upcoming year. Africa is another place where we're hoping to grow in the future because we really think this can have an impact worldwide on the scientific community. Now, it did start off with a focus on plant breeding, and I know this symposium has that focus as well. But we've really diversified through the years, and it was, what started off in plant breeding really grew into more diverse plant sciences. And now, as you noticed, I've kind of haven't really mentioned it, but our branding has changed a little bit. We're shifting away from the Plant Sciences Symposia series and moving towards the Corteva Symposia series, mainly because we think we can be even broader than the plant sciences. And I'll show you in a moment an example of that. But yesterday and today, this is an example of exactly how this should work. Students coming together and really providing a really interesting group of speakers and a topic of interest to both their graduate program, students around the world, and faculty. So I think this is just a fantastic example. And the Plant Science Council really, I think, exemplify the leadership and these skills that we are really going for, and not just through the Symposia series. I know the Plant Science Council is really active through the year in programming and developing, providing opportunities for students. And so, Josh and Sudip and all of the participants in the Plant Science Council, I'm really proud of what you do through the year. And this is just one example, the Symposia series, but it's a great opportunity for me to really interact with all of you. And so I'm really thrilled to be a part of it. Normally, this is what we would be kind of looking like at the University of Florida, gathered together in a large lecture hall and interacting in person. But obviously that we were hoping to get back to a little bit more of that in-person component this year, but obviously things are beyond our control in many ways. And despite the fact that we've been virtual since for almost 2 years now, for the most part, I think the virtual symposia have really had a big impact with really large numbers of participants around the world. And so I think the real challenge this year is yes, we will get back to in-person events. I hope to be in Gainesville next year, but we will we will be driving towards that. But I think the real challenge that's coming is how do we keep this virtual impact in the real world? You know, we want to get back to being able to meet together and network together and really face to face. But we don't want to lose the global impact of the things that we're doing. So that will be the real challenge facing students moving forward. How do we effectively do a hybrid event? And I dare say, I think many of you know that hybrid events are more difficult than in-person or virtual events in many ways. So I think that's a challenge facing a lot of conferences now, as well as these symposia. So I'm excited to see what students are going to come up with. But we had a great year last year. We had a record 39 events. It was really a successful year. We had some really innovative programs. As I mentioned, we're not just the plant sciences anymore. We had our first chemistry symposium, which was co-hosted by the University of Michigan and Yale University. It was a really exciting event that I think opened some eyes, and really, I think it's important that the agricultural industry kind of has a better visibility in some of these scientific disciplines that maybe they may be looking to pharma or other places. So that's one example there. We had some really innovative programs in Brazil last year. We had a seed-thon, which had competition between many teams across Brazil and solving problems in the seed industry. So my group of Brazilian students down there, I'm always love to see what new and interesting things they come up with. So I'm looking forward to a great year. Obviously, this is our kickoff, our first one here at the University of Florida. We have Missouri coming up in February, as well as Texas A&M. And I also want to point out we have an HBCU Ag conference at the same time, kind of overlapping with Texas a little bit there, but maybe many of you are adept at having your laptop and your tablet or something running at the same time, and maybe attending two conferences at the same time. But I'm really excited about that. That's actually Florida A&M is going to participate in that. It's actually 4 different HBCUs: Tennessee State, Kentucky State, Alabama A&M, and Florida A&M. So it's a great group of students putting together an ag-oriented symposium in February. So keep your eyes open for that. Hopefully our webpage will be updating soon, but I will share links with organizers who can then send those out. And if you ever have any questions or you want to be connected, please feel free to reach out to me. Oh yeah, here's the advertisement, the brochure for that upcoming HBCU event. So last thing I want to leave you with here is my open innovation group at Corteva is really looking for opportunities to reach out and collaborate with the external academic community and small companies and things like that. So we do that in many different ways. We respond to requests for, we have opportunities to respond to requests for proposals, so collaboration on specific topics. We have crowdsourcing challenges, and we also have the opportunity to just submit a proposal or idea at any time. So this is the way to do it. If you have an idea and you think that might be a good partner in that idea, so we have some really great opportunities for that protects your IP, and I think works well for everyone. A couple proposals out there that we have right now. We have February 15th submission deadline on a genome editing for broad spectrum disease resistance in crop plants. So if that's something you're interested in, please check it out. You get, if you were awarded this, that your proposal gets awarded, you get vector design and synthesis, scientific consultation, and you receive your gene edited seed. So exciting opportunity there. And then we have others in if you have friends working in small molecules and in crop protection might be interesting, we have another one there. So check that out. That's at our open innovation page. But if you have any questions, please reach out to me. There's my email address, connect to me on LinkedIn. And I'm looking forward to our second day of the symposium here. And I will pass it back to the organizers. Thank you very much.
Thank you, Dr. Joshua, sir, for your encouraging opening remarks. I would also like to thank Corteva AgriScience for their continuous support in academia and for grad students to organize all these kind of events. Now I'll hand the floor over to Nadia Mourad Silva, the Plant Science Council Outreach Chair, who will moderate for our next speaker. Nadia.
Good morning, everybody. I'm happy to introduce our first speaker today. Dr. Kevin Bexey is an assistant professor in the Department of Environmental Horticulture at the University of Florida. His research group focuses on understanding plant developmental responses to biotic and abiotic stress. His overall research goal is to elucidate novel pathways and mechanisms that could be used to improve crop performance under unfavorable environmental conditions. I'm excited to hear about how this research relates to our overall theme of breeding for climate change. So Kevin, the floor is yours.
Thank you, Nadia, for the introduction. So let me share my screen with everyone. Can you see now?
Yes.
All right, perfect. First, I would like to congratulate the organizers of this fantastic symposium. I think it's a great opportunity for discussing great science and networking with different people in the industry as well as in academia. So today I'm going to be sharing a little bit of what my group have been doing the last probably 3-4 years, studying stress responses, particularly in maize. So I would like first to acknowledge the fantastic group of students that have been conducting this research. The data I'm going to be presenting today mostly comes from Shinli Li. He's a 4-year PhD student. Andrea Geza, he's a 2nd-year PhD student. And Katie Chase, she's a 1st-year master's student. So if we were in a in-person symposium and I asked the audience whether you have been stressed lately, I assume the entire crowd will raise their hands. So can you imagine what happened with plants where they are either in the wild or they are in the field, experiencing normal changes in temperatures, normal changes in continuous seasons, plus pathogens and diverse abiotics as well as biotic stresses. So how plants cope with these constant changes? So this is one of the topics of our research. And the approach that we take, we mainly are focused on reproductive development. So even though there are many important stages during plant development, we are interested in 3 main components. The female gametophyte is here, the male gametophyte, and the seed development. So the data I'm going to be sharing with you today mostly comes from our results in the male gametophyte. And for that, we have been using a particularly interesting crop, maize, as a model. Maize arguably is one of the most important crops globally. It has a really well not annotated, but well sequenced genome. Lately have been new updates on it. But during this climate change situation, we have seen that so we have seen that in the last probably 100 years, temperatures have fluctuated in a global scale. We have seen regions where temperatures become 1 or 2 degrees centigrade above the threshold. Others during the winter time becomes really cold. So we have seen this climate change varying across the entire planet. So that have imposed a particular challenge on crop production. So in order to see how those changes impact crop productivity, as I said before, we use maize. And this is the results of a picture of some of our plots in, I think it was 2017, where we saw a really dramatic decrease in yield. We see a few seeds in some crops, others we saw kind of halfway through. But then we were interested actually what is causing this particular phenotype. When we look at the male phenotype, this is something that many of you have seen, is that you will see pollen grains that are fully formed, which means that they are viable, the ones that are really dark here. And you see also pollen grains that are kind of unviable, the ones that are more light color here. So those particular pollen, they don't germinate. What that means is they are not able to transfer the genetic material from the male side to the female side during the fertilization process. So many studies conducted, this is something not new. We have seen that across many plant species. We see, for instance, examples in brassica, populus, beans, where heat stress basically impacts pollen development. This is something that have been discussed for many long times. Scientists, even in 80s, 90s, they already realized that particular problem. Even though this is not novel, the research in my lab takes a different approach. So rather than looking at the broad context, how heat stress impacts the entire overall reproductive development, we are interested to see how are the impacts in every single developmental stage. And the reason why we are looking at developmental stage responses is because we know that there are tissue-specific gene expression, tissue-specific protein expressions. We see that every developmental stage is different. So in order to see how plants respond at the developmental level, we wanted to look at how each pollen stage have been impacted or is impacted by heat stress. So here, this is a normal pollen development transition. So it starts with pollen mother cells, which then goes to myocytes, then tetra stage, unicellular, bicellular, tricellular, and then mature pollen, which is the one that germinates and then fertilize the ovaries. So this can be divided in microsporogenesis and microgametogenesis. One of the main problems in studying developmentally the impact of stresses is how you are able to dissect only the developmental responses rather than the entire response of the plant. So in order to do that, we first develop a method that allows to systematically isolate pools of individual pollen stages during maize plant development. So we use the well-known leaf color method, and we try to correlate it with the different stages of pollen development. Even though we didn't find a 100% correlation, we were confident that the amount of pollen that we can isolate it were from an individual pool or individual developmental stage. This particular study was conducted in B73 Imbrine line. We tried W22, A188. The results are not as consistent as B73, but that's something that we can play around, and that can give us good number of isolates in individual developmental stages. So for instance, you can see here in V13 that most of the pollen is in the unicellular stage. Again, we use the central spike of the tassel, so we were just concentrated to have a unique pool of cells in the same developmental stage. And you can see here that basically most of the cells that we isolated were in the same developmental stage. Here in the tetra stage, which means we use mostly V12, actually V12 maize plants, and here we use, this is bicellular stage, and we use mostly B15 plants. So after we have developed that method, we went and then look at every individual developmental stage. I will be sharing mostly two stages, tetra stage and bicellular stage, even though we have conducted research on almost all the stages. Because of time, I'm going to be sharing the only tetra stage and bicellular stage. So bicellular stage is a really important stage during pollen development, since it's a transition between the microsporocyte to the megasporocyte. So one thing that happens before that transition is that at the tetra stage, you can see here, there is no starch accumulation. So this is previous starch accumulation in the pollen development. Why starch is important for pollen development? Because that's the one that basically going to produce, helps with the energy production for later germination. So we particularly focus on heat stressing plants that were at the tetrad stage of pollen development. Here is just a scheme showing how we did that particular study. We heat stressed it here at the tetrad stage. You can see in the red line. And we also kept a group of plants as a control, and you can see here in blue at normal temperature. We conducted physiological, biochemical, omics technologies to see how actually the plants responded. And we collected samples right after the stress, during the stress, as well as later during normal development. Something that I wanted to point out is that after we applied the heat stress, which basically lasted for 2 days at 35 degrees, we placed the plants back into normal conditions just to see how that particular stress only during the tetrad stage may have any impacts on normal pollen development. To our surprise, that transient stress of 2 days impacted greatly the shape of the tetrad stage. You can see here on the top, non-stress pollen, you see the round type of shape of the tetrad stage. However, the heat stress ones, they clearly were a little bit shrink, and that one was something that caught our attention. Since we know that pollens are not individual entities and they are maintained in capsule within enters, we also look at an enter development. Even though enter development is something that have been really well studied, we just wanted to see whether the stress that we applied actually caused similar phenotype I've described before. And we look at here at the tetrad stage, maculated microspores, bicellular stage, tricellular, and mature tricellular pollen. And here on the top, we have non-stress conditions. On the bottom here, we have heat stress conditions. And 3 important components stood out that were different. So we see here that the epidermis cell layer was degraded a little bit slower in the heat stress samples than the non-stress ones. That caused a little bit of delay of the releasing of the pollen. Also, we observed a degeneration of the tapetum. It was a little bit slower again on the heat stress samples, as well as endocytium. It is interesting because even though we apply heat stress, many have reported that the tapetum degrades faster, at least in maize and in the Imran line B73. This particularly transient heat stress actually delays the process of tapetum degradation, which is really important for the pollen formation. Another important observation was that you see here the mature tricellular pollen in non-stress one was really strong, a strongly intense stain, which is an indication of pollen viability, something that we didn't observe here in the heat stress ones. So after we analyze right after the tetrad stage, and when we stop the stress and then we put the plants on their normal development, we look at one important trait for pollen, which is pollen germination. So first we did an in vitro assay, which basically consists on putting the pollens on a media that allows pollen to germinate. We provide all the components needed for the pollen to germinate as if they were in the plant. What we see this in vitro assay shows that many of the heat stress pollen didn't germinate, as you can see here in the white arrows. A few, as normal, in the non-stress one also didn't germinate. So when we quantified that particular response, we observed that around 80% of the non-stress pollen germinated, and around 20% of the heat stress ones also germinated. For the people that work with maize, you will say, okay, 20% of pollen in maize, because of maize has a lot of pollen, that maybe doesn't have a really strong impact on fertility or seed set. But before moving to that, we also did an in vitro assay. So in vitro assay, basically we use normal silks. We place non-stress pollen and heat stress pollen. And in the non-stress one, we see how the pollen tube germinates normally. So basically germinates penetrates the silks and basically goes all the way down to reach the ovule. In the heat stress ones, the pollen is able to stick, a required process for pollen germination. But even though it is able to stick into the maize silks, the germination didn't happen. So we went further and analyzed to see whether the effects of seed set. And what we see here is our controls. So basically non-stress pollen and non-stress cups, we see full seed sets. And basically here we use non-stress cups and heat stress pollen. And we see a diverse type of response. Some cups were more full than others, but again, in general, we see a strong reduction in seed set, basically because of the failure in fertilization. So we wanted to understand better what was the cause of this particular phenotype. And then we started biochemically checking on pollen, mature pollen after being stressed at the tetra stage. And we first look for a starch, which is one of the most important components, as I said before, for pollen germination. And this is a non-stress pollen, and this is a heat stress pollen. You see partial accumulation of cellular components. So basically starch were just reduced. And when we quantify it, basically we see a significant reduction between non-stress and heat stress ones. Same happens for a technique called FDA staining. FDA is a enzymatic assay that allows a chemical component called FDA to penetrate the pollen. When the enzymatic activity is really strong, you see a strong signal, as we can see here, fluorescent signal. When the enzymatic activity is low, basically you see a decrease in the fluorescence. When we quantify, also we saw a significant difference between the heat stress one and the non-stress one. So this one was the first indication that the starch as well as enzymatic activities were impacted. So we went and did a transcriptomic approach. We use RNA-seq Illumina analysis. As you can see, we found many genes that were disrupted by the heat stress at the tetra stage. And again, I would like to reinforce that these particular samples were took at the maturity. So first we use cellular markers as expected, heat shock proteins, which are important for markers as the heat stress markers. We observed that even though the stress happened in the tetra stage during the recovery process, those genes were still upregulated. So even though the stress happened before, at least this particular inbred line was not able to recover again to the normal stage. We see a really high expression of those particular markers. So then we look at exactly what was the responses, what happened, why this particular pollen didn't recover. And we found a lot of genes that are important for fertilization. For instance, kinases are known to be important for the fertilization process. We study a few of them. We saw upregulation and downregulation. And then we perform a network analysis, and basically a few of them work together with energy pathways as well as the heat shock protein responses. So we went further and look at exactly at the biochemical pathway that were impacted. And among the different ones that were impacted, we look at two main important ones for the pollen germination, which are the energy biosynthesis pathway and the lipid biosynthesis pathway. As you can see here, the enzymes, or also the genes in red, are upregulated. The ones that are in blue are downregulated. So first, we're going to be looking at this particular energy biosynthesis pathway. And what we saw was that even though, as you could remember, the starch was reduced, and the enzyme that basically converts starch into glucose also was reduced, ADP glucose. However, plants didn't basically stood there. So in order to overcome this particular reduction in the amount of starch, maize pollen use another pathway. So they use the invertase, which basically converts glucose into sucrose into glucose and fructose. So this, even though this particular pathway was diminished, plants basically turn all the hope into the invertage to basically be able to produce enough energy. And also was another gene and enzyme were upregulated, the phosphofructokinase. So this one was shut down, but this one was increased here. However, at the end of the pathway, we saw also a reduction. Phosphate dehydrogenase as well as enolase, important for the conversion of this particular glyceraldehyde into pyruvate. They were downregulated. What we observed was at the end, ATP synthesis was decreased in the heat stress pollen compared to the non-stress one. So in order to prove that not only at the transcriptomic level, that was the case, we performed metabolomics, and we actually confirmed our transcriptomic results. So glucose is upregulated, was upregulated here in heat stress samples, and this upregulation basically comes from the invertase activity, not because of the ADP glucose activity. Same, fructose was upregulated, as we can see here, both. And then pyruvate levels were slightly reduced in the heat stress compared to the non-stress. A similar observation was in the lipid biosynthesis pathway, where basically lipids are really important because they are stationary components on the newly membrane formation during the pollen tube growth. We observe upregulation of the 3-oxacyl synthase as well as the glossy 1. However, acyl carrier desaturase was downregulated. So our hypothesis is that this particular gene was the bottleneck, even though those was upregulated. This particular gene was not able to convert to the kinol into the different lipid compounds. And basically, when we perform metabolomics, we observe a reduction in the heat stress samples in all the different lipids with different carbon compounds, C15, C16, C17, and C18, which are all of them fundamental again for the cell wall formation, the cell membrane formation. As a model, we propose that basically when heat stress happens at the delta stage, we see an increase in sucrose because of the activity of the invertase, which increase the excess phosphate pools. However, the starch, which is also another contributor of the phosphate pools, basically was decreased, and therefore because of the activity of the ADP glucose. That one resulted in decreasing pyruvate, fatty acids, and energy, which basically impacted the pollen germination as well as fertilization. So that's one initial response that we observe at the tetrad stage. So heat stress at the tetrad stage of pollen development causes male sterility. And this is what basically because of two important, the heat stress targeted two important biochemically important pathways, energy and lipids. But then we shifted our attention to the bicellular stage. We were expecting similar results, and we used the same leaf color method as a way to collect the pollen. And one thing that actually took our attention was that the heat stress pollen at the bicellular stage didn't respond as expected. We didn't see any effects in the pollen germination when they were mature. So we did in vitro assays, and we also did in vivo assays. You can see the pollen grains germinating normally here in the controls or non-stress samples versus the heat stress ones. Same happens here in vitro. So this is something we repeated multiple times, getting the same results. We then decided to look at more in detail. And here you can see starch composition. They were no different. We used DAPI staining. Basically, DAPI is a staining that allows coloration of the DNA within the nucleus. Those were in the same developmental stage. We didn't see FDA staining, which was no difference between heat stress and non-stress. We use another staining, Alexander staining, which also stain for starch. We didn't see differences. We check, okay, if the difference are not, maybe the germination speed of the pollen tube is different. No, we didn't see differences in germination, in the speed of the germination of the pollen tube. Also, the pollen germination, when we quantified, we didn't see any difference at all. Same for pollen tube length. So at this point, we were, okay, maybe bicellular stage is resilient to stress. So when plant development already reached that stage, is not relevant when the heat stress happens. Maybe the earliest stages are more susceptible. And then what we did, we just cross heat stress pollen at the bicellular stage with non-stress cups. This is our control samples, full seed set. But again, we observed the same similar phenotype. So that was a really contradictory result, because if we didn't see anything happening here within the pollen and the female side was not stressed, why we still see a fail in the fertilization? So in order to look at and try to understand that particular observation, we study, we propose we look at a different hypothesis. So again, this is a normal pollen development stage. We have tetrad, unicellular, bicellular, and tricellular. One of the important stages between the bicellular to the tricellular is the formation of the sperm cells. This is the vegetative nucleus, this is the regenerative nucleus, and then after this transition, they separate, forming the two sperm cells. One is gonna fertilize the embryo, the other one is gonna fertilize the central cell to form the embryo, the other one is gonna fertilize, gonna form the endosperm. So our hypothesis was that basically there was no formation of a second sperm cell. So basically there was no separation of the regenerative nucleus forming the two sperm cells. And that's why basically there was no fertilization at all. That's basically explained here, where after heat stress, you need to have a formation of two sperm cells, which need to travel and then fertilize. Our hypothesis was that only impair pollen travel, and because they were not two particular sperm cells developed, we observe a failure in fertilization. In order to confirm our hypothesis, we constructed a marker line. So we used a YFP marker line. We knew that we have a few genes that are only expressed in the sperm cells, and we fusion that particular gene with the YFP to visualize the particular expression. When we had that plans ready, we perform a heat stress experiment. On the non-stress one, we see here, this is basically the sperm cells. You see a nice spindle formation. So after the bicellular to the tricellular, you see a division of these particular sperm cells, forming two individual sperm cells. Clearly here, this is the intermediate stage. In the heat stress ones, we didn't see that particular clearly. It was kind of affected that process of separation of the sperm cell formation. So, okay, that one match a little bit with our hypothesis. However, when we look at in detail during the pollen germination, we observe that indeed the sperm cells, they are able to travel, and we observed two of them. So our initial hypothesis that the sperm cell formation was impacted was actually not true. What actually happened was that the heat stress samples kept the sperm cells inside the grain. So the genetic material never traveled out into the pollen tube. That's why the fertilization was impaired. There was no genetic material traveling to the pollen tube, so the fertilization process was particularly not happening. Nothing in contrast with the non-stress one, where you can see here the two clearly sperm cells traveling across the pollen tube. So in order to understand better that, we isolated sperm cells. So we use a technique called a Perkel gradient. So we use different concentration of Perkel to isolate only sperm cells. Many studies have been conducted with pollen, but they use entire grains, which basically didn't does not provide information regarding specificity in the sperm cells or the entire pollen grain. So we decided to do only sperm cells. We isolate individual sperm cells, as you can see them here. And then we perform again a transcriptomic analysis. And we observe around under heat stress at the bacillary stage. We observe around 600, almost 700 genes totally misregulated. Around 478 went up, around 200 were downregulated. Here is just a heat map showing a few genes up, a few genes down. We observe, we perform gene ontology analysis, many genes up, many genes down. At this point, and as you may imagine, many of the studies base their decision on designing or to study candidate genes on only transcriptomics. So we didn't want to base our decision only on transcriptomics. So we also performed sperm cell proteomic. What we found was that there were around 1,400 proteins that were common on both, and around 600 were unique for non-heated, non-stressed pollen, and around 1,300 were unique in the heated stress pollen. So we crossed the proteomics with the transcriptomics, and then what we did, we picked samples, genes that were present in both data sets, not only in the transcriptomic, but also in the proteomics. Since maize transformation takes quite a long time, and we didn't want to risk of studying genes that maybe are not the target of the heat stress, or genes that were not actually giving the response of what happened during the sperm cell. We first perform in situ hybridization. So we studied initially 2 genes, and the in situ hybridization allows us to basically confirm where the stack expression of those particular genes within any given tissue. When we look at the enters, we see a clear expression, as you can see, a strong signal here in the sperm cells of both the gene 1 and the gene 2 target. Then we perform also transient expressions. First, we use tobacco pollen transient transformation to see whether those genes were indeed expressed in the sperm cells. We see strong expression in the here in the sperm cells of tobacco pollen tube, as well as in the pollen grains. We also did transient transformation in leaves of tobacco plants, and they were again strongly expressed in the nucleus, an extra confirmation. But then we also use BMS suspension cells, again, to double check that those particular genes are expressed into the nucleus. We wanted to be sure before embarking in a transformation process to be sure that our genes were actually the targets. So we see a strong expression again in the nucleus. So we decided to go further, and we perform CRISPR. We designed CRISPR for those 2 genes. We use 2 different set of constructs. We use in the same construct, we target both genes in order to maximize the chances to have a double mutant of both genes. And the reason why we did it with both genes, because those genes were genes belonging to the same gene family. What we found a myriad of different transformants. We found single mutants. We found most of the transformants that we got were single mutants. So we started the process of crosses. Interestingly, we found genes that plants that were edited with around 50 base pairs, and another plants were edited around almost 900 base pairs. So we got plenty of plants. We started genotyping all of them, and this is how they look like. During vegetative development, they look normal, nothing different than any wild type plant. So at this point, what we are in the T1 generation. And this is just wild type transform, and these are the 3 independent CRISPR-Cas9s. What is important, we have a heterozygous of double mutants, and what you can see here is that at the tricellular stage, you will expect to see the 2 sperm cells and the vegetative nucleus. And see here, you can see here the 2 sperm cells, vegetative nucleus. Something that is really interesting for us right now, we observe, you see strong signaling all over. So the function of that gene is involved in several components during the replication process. And interestingly, the sperm cells are not clearly formed. So basically, you don't see a particular condensed sperm cell formation. You see a dispersed signaling all over. So our hypothesis initially is that maybe that particular 2 candidates are important for the sperm cell architecture, for the chromatin architecture, and that's why they are just basically, when they are not present, the structure cannot be maintained, and then the genetic material is dispersed all over the pollen grain. We also look at pollen germination, and you can see wild type transform. You can see here the normal germination of the pollen pollen tubes. However, in the mutants, the double mutants, what we observed was that they start to basically not growing as here. You see straight pollen trying to find, the ovule. Here they start to growth, and then they start to basically curl a little bit, and then it is a really strange change in orientation, something that is not normally observed in the wild type pollen. So now we are quantifying the differences between that particular phenotype to see whether this is something that is related to the 2 genes that we were looking at or something else. And then we also look at the fertilization rate, C-set, and we cross them with B73, we cross them with A188, we are also crossing them with W22. This is here the wild type. We have started seeing a few reductions in some of the crosses. However, this is not conclusive, since still we are working with heterozygous plants, and they are in the T1. We are in the process of getting homozygous plants in the T2 generation. After we confirm that they are homozygous, we're going to perform the heat stress analysis and whether those particular 2 genes are critical for the response to the heat stress, as well as the sperm cell formation. So in summary, and what is the take-home message is that the impact of heat stress on pollen development is different depending on the stage. So it's not the same that when you see a stress happening, if we go to the vegetative development, what happened during the seedling stage, that during before flowering transition. So every developmental stage is different and has a myriad of responses. So in the case of pollen development, is different depending on the stage. So the third stage is susceptible, and basically the susceptibility comes from the impact on lipids and energy biosynthesis. The bicellular stage is a little bit more resilient, seems to be, but then at the fertilization stage, the fertilization is really impacted, which means that the responses or the molecular pathway that are affected by heat stress at the bicellular stage are different than the tetra stage. So with that, I would like to thank everyone, and I will be happy to take any questions.
Thank you, Dr. Bexy. We have a question from the chat. From the results of your work, we can see that high temperature affects pollen negatively and hence causes significant yield decrease. In the current climate change scenario, do you think that the development of heat tolerant lines using some of these genes that you were working on for pollen development should be one of the main focuses for maize breeders?
That's one of our objectives. So we want to find critical genes that are help breeders to have more resilient pollen. So pollen that, even though heat stress happens, there is enough pollen for the seed production. I didn't show here, but we have also other candidates that basically the amount of pollen that they produce is a seed way more controlled. So basically we have twice amount of pollen than the control plants. So that has a really nice potential implication, because if you increase the amount of pollen, even though the stress happening, a good amount of them gonna have good chance to fertilize properly, and therefore you're gonna have a normal seed set. So we are working on those candidates to see whether they perform really well in the field. So far, the data we have is data in the greenhouse, but we are now transitioning to the field to see whether the performance is as observed in the greenhouse.
Thank you. I have another question from Dr. Narakuruti. She asks, what is the hypothesis for the slow degradation of the tapetum and the epidermal layer under heat stress? Why do you think? Yeah,
that was the first study, right? So in this case, what we think is that because of our stress was only for 2 days, it was not long enough to cause what many people have observed, a rapid degradation. So I think these two stress, basically what it does is just mess up entirely with the molecular responses, and then the plant didn't know exactly what to do. And basically what happens, the tapetum started to slowly degrade, just to release, just to acclimate to the stress conditions, and that's what we see the differences.
do you think that the disruption of those cellular structures, the tapetum and the epidermal layer, also contributed to them not being able to germinate well?
That's really an important stage. So the tapetum is known that basically nourish the pollen. So there is a critical developmental transition where the tapetum start to release all the compounds that will nourish the pollen. If that nourishment process is affected, as you may imagine, for instance, what happened with small babies, if they are not properly nourished, the normal development is going to be impacted. So same happens with pollen. If the normal nourish process is affected, the pollen doesn't want to get the biochemical and physiological, also the molecular response that is normal expected during pollen development.
One last question that I have you considered doing overexpression lines so that on those replication genes that you spoke about, maybe this will create more pollen?
That's something, the one that I talked, we have overexpression. So basically the one that produces more pollen is our overexpression line. The one that has have a CRISPR, the gene that have been CRISPR basically reduces the amount of pollen. So we see a really dramatic change when the gene is overexpressed and when the gene is not present. So it's a really promising, but still we are in the data in the preliminary stage where what we saw was only in the greenhouse. We want to be sure that it actually happens in the field.
Thank you, Dr. Bexia. I really enjoyed your talk. Those pictures and the videos are super cool. Very, very nice. I will now we'll take a short break. And when we come back, we'll have the 3-minute flash talk competitions with students. So everybody should expect to come back around 11. 11 a.m. So we'll have a 15-minute coffee break. Thank you, everybody. Dr. Bexy, somebody in the chat is asking for you to share the PowerPoint files.
Well, I can share a few, the ones that have been published, the one that not, because we are still in the process now, but I can share the ones that, I can share something with everyone.
Thank you. Or message Charles Ologidi directly.
Perfect.
Yeah, and let us know if you don't want this on YouTube, because it will be there unless you tell us otherwise.
Yeah, that's fine. Later we can talk about it.
Hey, Jordan, can you hear me?
Yep, loud and clear. Sounds good. Thank you.
All right. Just one question. I will share my screen, or are you gonna share the presentation?
So I will be sharing my screen and I've got all the slides compiled. And so I'll just be cycling through those. So you don't have to worry about that.
All right, sounds good. Thank you.
All right, no problem. Thanks.
Hey Jordan, can you hear me as well?
Yep, sounds good. Thank you, Swati.
Thank you.
Hi, Jordan.
Hi, sounds good. Thank you.
Okay, cool. Thank you.
Hey Jordan, can you hear me? Just checking my
Yep,
microphone. Okay. And you
sounds
can
good.
also
Thank
see
you.
me, right?
Yep, can see you
Yeah,
too.
sounds
Yep,
good. Okay, see you in a minute.
sounds good. Thanks.
Pikasani, can you try your mic again?
Hi, am I audible now?
Yes, loud and clear.
Yeah, thank you so much.
Hi, Jordan.
I can see him loud and clear. Video
Good.
looks good.
Okay.
All right, looks like all our speakers are here and panelists. I think we're just waiting on Pablo. All right, everybody, it's 11 a.m. now. Let's just give it maybe 2 more minutes, wait for those final judges to get into the Zoom. All right. Hello, everyone, and welcome back. I hope everybody had a good little break. Everybody was able to get their coffee or tea. So just to introduce myself real quick, my name is Jordan McBreen, and I'm a PhD student here in the agronomy department, as well as this year's 3-minute Flash Talk competition chair. So just a bit of a background on the 3-minute Flash Talk competition. The idea is that an effective scientist should really be able to deliver a summarized version of their research in only 3 minutes or less, and that being able to successfully do so really indicates a good scientific communicator, which is key in bridging the gap between research results and their real-world applications, be it in the field or in the consumer marketplace. Effective scientific communication also allows us to productively collaborate with one another by increasing our understanding of each other's works. So let's briefly go over the rules, and then we'll jump right into the presentations. There will be 3 judges who will be impartially scoring each of the presentations. The presentations are limited to 3 minutes maximum, and competitors exceeding these 3 minutes will be penalized with, and I'll be keeping the official time. Time begins when the competitor begins speaking. A single static PowerPoint is permitted. All questions will be saved for a separate time slot for all presenters to respond. And once all the presenters have gone ahead and presented, we'll go ahead and do the Q&A. So if anybody has any questions, be sure to put those in the Q&A as you're thinking of them. And then finally, there will be a 3rd, 2nd, and 1st place winner, with there being cash prizes of $100, $150, and $200 respectively. So be sure to do your best, guys. All right, and so to start off the competition, we have Andrew Biswas. Let me go ahead and I will share my screen, and then Andrew, you can go ahead and take it away. All right, I'll go ahead and I'll start the timer whenever you start talking, Andrew.
Hi, Jordan. Can you hear me?
Loud and clear.
So you want me to share my screen, or you are sharing the slide? I will go ahead with there.
So I'll go ahead and I will share all the slides.
Okay, sounds
Are the slides
perfect.
up there? Everybody's able to see?
Jordan, you're in presenter mode.
Yeah, it's like little I can see.
Oh, okay, okay.
Yeah, I think it's better now, but it's not slide mode, I guess.
Go ahead and start it. Start the slideshow. And then
how about that?
there we
Yeah,
go.
now you have to go to my presentation,
All
I
right,
guess.
okay. All right, okay. Go ahead whenever you're ready, Andrew.
Good morning, everyone. I'm Anju Viswas, a PhD candidate from Agronomy Department at University of Florida. I'm excited to share my research entitled High Throughput Approach for Predicting Alfalfa Yield. Predicting alfalfa yield depends on phenotyping. Phenotyping is very crucial in plant breeding. So what you can see from figure 1A, manual phenotyping, which is time and labor intensive. And you can see even my PI also in blue t-shirt helped us there. On the other hand, I am the only person in figure 1B accomplished the same task within 15 minutes. Isn't it cool? So high throughput phenotyping can be an alternative of manual phenotyping. So what's our research objectives? One of our objectives was to phenotype the alfalfa breeding population for harvest accumulation using both manual and using unmanned aerial vehicle. The second objective was to find out the correlation and prediction efficacy of high throughput phenotyping with manually collected harvest accumulation. Our research was conducted in Sitra, 30 minutes away from Gainesville, and experimental design was row column design. We had 145 full sieve and 36 half sieve families, including 3 controls. 4 manual harvests had been done in different time points in 2018 and 2019 based upon 10% flowering of controls. An unmanned aerial vehicle equipped with multispectral camera was flown before each harvest. Then the image processing was done in EasySoft, which is visible from figure 2. We stitched the image using EasySoft and QGIS to generate 4 vegetation indices based on 2 or more bands designed to enhance the contribution of vegetation properties. And figure 2 represents the workflow had been done in that software. Pearson correlation and linear regression was done in R later on. In result section, I'm presenting the result from one harvest as we found similar trends for all 4 harvests. You can see from figure 3, correlation between vegetation indices and harvest accumulation. The x-axis represents 4 vegetation indices and y-axis represents the harvest accumulation. And we found higher correlation, which is more than 80%, and medium prediction efficacy, which is visible from figure 4, which is more than 65% accurate between harvest accumulation and vegetation indices, which you can see already aside figure 4, where x-axis represents harvest accumulation and y-axis represent vegetation indices. So what's our take-home message from this research? High throughput phenotyping can be an alternative of manual phenotyping to speed up the selection process of alfalfa breeding population, and we will be benefited from that in our breeding program. Thank you, everybody. Thank you for listening to me with patience. I will be happy to take any question. Thank you.
All right, great job, Andrew. So next up, we have Julianne Janori. Julianne, the floor is yours.
All right. How many of you guys have actually taken a walk through a scenic downtown or through your local park and actually paid attention to the ornamental plants around you? Chances are you've overlooked one of the most common choices for beautifying these areas. And that plant, as seen in the image in the top left, is Begonia semperflorens, or the wax begonia. These beautiful ornamentals are typically used to line flower beds that line roads, walkways, or home gardens, but they're actually currently encountering some setbacks. Much of the wax begonia production in the US occurs in Florida, yet the hot summers coupled with intense light are actually taking a toll on the growth, leading to a loss of product and revenue for big growers across the state. If you view the panel on the left, you'll see that I conducted an experiment to first characterize the response of various wax begonia varieties to heat and light stress, and second, to see if there are any morphological characteristics that enhance the tolerance of any of the varieties studied. If so, these morphological markers will allow us to easily make beneficial selections in breeding. Now, when it comes to wax begonia, there are certain morphological changes that are uniform across all varieties studied. The stomatal density consistently increases when plants are exposed to heat and light stress compared to them being grown in the shade. As shown in the circular images on the left, the genus Begonia is unique because some species in the genus have stomatal clusters as opposed to a singular stomatal, which aid in gas exchange. Now, there's no actual change in the stomatal cluster sizes in either of the treatment groups. So now here's where it gets interesting. As I direct your focus to the middle of the slide, one of the traits that does vary depending on the species is the degree of leaf folding. Typically, plants fold their leaves to reduce the intensity of direct sunlight, and that looks to be the case here, but the intensity of that folding varies from variety to variety. When plants are exposed to direct light as compared to shade, there's a significant difference in that folding. Highlighted in yellow, you can actually see that one of the varieties, FB059, folded significantly more than the other three tested. The commercial green variety, denoted as CG and highlighted in red, barely folded at all there. Another key morphological trait that could perhaps increase the resistance to light and heat is the cuticle thickness. The story is the same as leaf angle, with FB059 not only having a thicker cuticle, but also increasing its cuticle thickness significantly more than any of the other varieties tested. So this is visible in the table highlighted in yellow and in the chart at the bottom. So now that we've identified morphological characters that vary from variety to variety, the question is, how do we actually know that they enhance heat tolerance or light tolerance or any of these abiotic factors? In addition to the morphological characters, I also wanted to know the physiological response of these varieties. As seen in the chart on the right, the ion leakage of FB059 in the unshaded environment is significantly lower than the other varieties. When looking at fluorescence, a great indicator of the stress level of the plant, we also see that FB059 has a higher FV over FM ratio, signifying that the plants are under less stress. The goal for this information is that it can be applied to making breeding decisions to improve the growth of wax begonia in the industry. Thank you.
All right, thanks so much, Julian. Great job. So next up, we have Kara Gerline. So you can go ahead and start when you're ready.
Hi, can you hear me?
Yep, loud and clear.
Okay, great. Hi, everyone. My name is Gerline Carr, and I'm a PhD candidate in CLE Lab here at the University of Florida. So we all like tomatoes with a good flavor, either in salad or a salsa. But as tomatoes have gotten larger, they got more water and have become nearly tasteless, with decades of breeding for producer priority traits such as fruit size, appearance, and disease resistance. In contrast, consumer prioritizes the flavor, aroma, nutrition, and sustainability. So there is a need to bring the lost flavor back in modern tomatoes for the consumers. As seen in Figure 2, tomato flavor includes specific amounts of sugars, acids, and a unique aroma, which is composed of over 400 chemical compounds that makes the flavor a complex and an expensive trait for breeding. When you chew a tomato, the flavor goes to the throat and to the olfactory nerves in your brain, and that's where you taste the flavor. Only sugars and acids will lead to a bland tomato. Most of the current tomato plants have lost the chemical compounds responsible for good tomato flavor. So now we are looking for ways to make big tomatoes more flavorful by combining them with diverse heirlooms, as you can see in the figure. My research focus is to identify and validate the genes responsible for flavor chemical compounds. To achieve this, I have used an integrative approach using multiple tools based on genome, transcriptome, and phenotype for flavor compounds. To put all pieces together for the flavor puzzle, we use a variety of genomic tools to identify consensus significant genes responsible for the synthesis of flavor compounds. The purpose of using multiple approaches is to better understand molecular breeding tools related to flavor. I have identified genes associated with flavor compounds, and currently I'm working on gene editing to validate the gene function. So the overall implication of my research work is to unravel significant genes critical to improve tomato flavor, and that could be a step closer to make big tomatoes taste better for the sake of our consumers. Thank you very much for listening.
All right, great job. Thank you so much. So next up, we have Lise Porto.
Yes.
So go ahead whenever you're ready.
All right. So, hello everyone, I'm Liz Porto, and about 2 years ago, I've moved to Florida to pursue my master's. And since then, I've noticed the importance of having an umbrella. Because Florida's climate conditions is unique, presenting warm temperatures and high humidity, and these conditions are very attractive for disease development. And lettuce crop has many diseases affecting its production. However, the foremost important disease of lettuce is downy mildew, caused by Brimia lactuca. And this pathogen is very genetic diverse, presenting several races. And the race structure is defined by using a set of differential cultivars with known resistance or susceptible reaction to this pathogen. So the idea of this work was to use the same lettuce downy mildew differential cultivars to be tested with other lettuce diseases in order to find common resistance in the lettuce crop. So 3 different isolates of Xanthomonas hortorum petovarvitens, the pathogen of bacterial leaf spot, were tested, were inoculated in the 17 differential cultivars. And after 10 days, these plants were evaluated for disease severity using a rating scale varying 0 to 5. Non-parametric analyses were conducted based on the relative marginal effects. 2 cultivars, Muriatis and Sylvinas, had low disease severity similar to the resistant control. The same idea was applied to Fusarium wilt experiment, where the roots of the differential cultivars were merged into a suspension with Fusarium isolate, and weekly, these plants were assessed for root discoloration and foliar damage using a rating scale varying 0 to 5. And the relative marginal effects showed that 3 differential cultivars, Sylvinas, Balesta, and Colorado, presented even lower disease severity than the resistant control for both parameters analyzed. So my research suggests that identifying multiple disease resistance in a single accession is possible. And in a scenario of climate change, where resistant outbreaks are suspected to be more frequent, the identification of accessions with multiple resistance is key for breeding programs, since, unlike myself, lettuce plants cannot use umbrellas. Thank you.
All right, thank you so much, Lise. So next up, we have Lushan Gamir. Lushan, whenever you're ready.
For some reason, I'm not able to on my video.
Oh, that's okay. No worries.
I think it's good now. Okay. Hello, everyone. My name is Lushan Gimire, and I'm currently a PhD student on a PMCB program working with Dr. Patricia Munoz in Blueberry Breeding and Genomics Lab. So now moving towards my presentation, plant breeding programs, they need to be aware of current and emerging threats to ensure future food security, especially for perennial crop plants such as blueberries, for which breeding is time-intensive and resource-intensive. It is very critical to identify the regions in genome that are associated with desirable traits if we really want to accelerate the breeding process. Annual Florida Blueberry Growers Surveys for breeding trait priorities has ranked fungal disease anthracnose among the most important disease in terms of incidence. Not just decrease in production due to this disease, it has already decimated planting of commercial cultivar flicker, which is a common parent in many hybrids. Therefore, to deal with this issue, we aim to identify genomic regions associated with resistance against this disease. Now for this, a multi-parental population in a 4x4 half diallele design was built with a total of 700 individuals. And I'll briefly tell you about how a 4x4 half diallele design looks like. It is exactly, the design looks visually exactly how it is shown in the gray-white table here in the middle with 4 different parents. And one thing to note, all these parents exhibited different levels of resistance and susceptibility in the field. Now, For phenotyping post-pathogen inoculation, every individual was given a score by observing its disease severity as shown in the photo with leaves here below the table. And the same population was genotyped using capture sequencing. Now, this method of sequencing allows us to extract genomic information from the regions of our interest. Now, let's look at this graph to our bottom right here. Preliminary results from phenotyping showed that the crosses were segregating for the percentage of susceptible plants. Additionally, further analysis showed that some crosses were working better or worse than would be expected from them, which means that specific combining ability of each parent was significantly different. This has hinted us towards potential polygenic genetic architecture of this trait. Now, the next step will be to link phenotypic and genotypic data to identify those associated genomic regions. This way, we can understand the genetic architecture of this trait, accelerate the development of resistant cultivars, and minimize losses due to disease in blueberry fields. Interestingly, this pathogen has a broad host range, so resistant genomic regions found in blueberries also have the potential to be introduced into other host species as well. Thank you everyone for listening.
All right, thank you so much. So moving right along, next up we have Mithri Bikasani. Mithri, whenever you're ready, you can go ahead and start.
Yeah, I would like to start now and wish you all a great day ahead. And I'm Bikasani Maitri from Indian Council of Agricultural Research, Indian Institute of Millets Research, located in southern part of India in Telangana state, Hyderabad. And this ICAR-IIMR is one of the previous agricultural public research institutes in India, dealing with basic and strategic research exclusively on sorghum and other millets like pearl millet, proso millet, finger millet, foxtail millet, barnyard millet, kodo millet, and together they count for about 8 millets. So yeah, coming to my crop, that is proso millet, which is Panicum miliaceum, also called proso millet, broom corn millet, or even white millet in France. So this is domesticated in China years ago. And the crop has specific notable features like rich amount of protein among all the cultivated cereals, which is almost like 13 to 17% over wheat or rice. And then the crop has, the grains has almost very good energy, and the crop is also gaining importance as a biofuel crop, and the studies are yet to be out. And this crop especially has highest water use efficiency, that is highest harvestable yield per water use among all the cultivated cereals. So yeah, like until last year, the grass, which is Panicum virgatum, which is the closest relative to this crop. So till then we thought of using only the switchgrass genome sequence to filter out the SSR markers. So these markers are now mined out using the proso millet genome sequence, which is released only a year ago, and using the GMATA, that is genome-wide microsatellite analyzing toward application, wherein the sequence from NCBI is taken and inserted in this bioinformatic tool, and through which we can really filter out the desired SSR markers by applying various filters like the motif, number of repeat motifs, and number of base pair lengths, and finally we have come out with many SSRs, but out of which we have to really reduce it to 90 SSR markers based on various parameters like number of AT repeats and GC content among the markers that were out, and we have to really eliminate out primers for those loci which are very critical to design any primer, and only those with A and T and those for which the two loci are very close. So keeping in parameters Of all these together, we have filtered out 90 SSR markers, and we are really screening out them using 24 genotypes to check for their polymorphism. And yes, they're really working well. As you could see, the gel image of one of the markers with which we have run PCR using 24 genotypes. And ultimately, these are the first stage SSR markers that are out from the main reference genome of proso millet. And I hope these markers would definitely help in studying the genetic diversity studies of proso millet that are located worldwide. Thank you.
All right. Thanks so much, Mitri. Great job.
Yeah. Thank you.
So next up, we have Pablo Sipowicz. Pablo, if you're in here, if you can go ahead, then the floor is all yours.
Hi, can you hear me?
Yep, sounds good, Pablo.
Okay, perfect. Hello, my name is Pablo Sipowicz. I'm a student, I'm a PhD student at Dr. Rios' lab. I will present our studies in genetic diversity in wild and domesticated annual ryegrass. Annual ryegrass is the common name of the species Lodium multiflorum. It's mainly used as a forage crop and is self-incompatible. So when we describe accessions in annual ryegrass, we should think about highly heterogeneous populations. The University of Florida's ryegrass breeding program has successfully exploited this heterogeneity by releasing more than 20 cultivars in the last 20 years. However, in the recent years, there has been evidence of or signs of germplasm exhaust. And to tackle this issue, the breeding program has selected 139 accessions from the USDA's National Plant Germplasm System to try and broaden the genetic diversity. These accessions can be divided by their passport status in wild type, cultivated, and unknown. So the objectives of our studies were to assess the genetic diversity in the USDA's green collection, the University of Florida's breeding program, and some commercial cultivars. And for this, we genotyped the materials with single sequence repeat markers. In the new collection, 50 experimental lines from our breeding program and 13 commercial cultivars. And the data analysis indicates that there's a presence of population structure in the collection that we analyzed. And this is important because if the collection is going to be used for association studies to discover QDLs of interest, there should be a correction for the population structure to avoid bias. And data also suggests that the wild type accessions that were selected are genetically distant from the rest of the materials that were analyzed, indicating that the addition of the wild type accessions will definitely broaden the genetic diversity of the germplasm. And our future interest of research will be centered in developing a genomic selection scheme to integrate traits of interest in our breeding program. Thank you very much.
All right, great job. So moving right along, next up we have Kasim Ali. Kasim, whenever you're ready.
Can you hear me?
Yep, loud and clear.
Hello everyone, my name is Kasim and I'm a researcher in Dr. Alpeter's lab in agronomy department here at UF. Biodiesel is a renewable and environment-friendly source of energy, mostly derived from oilseed crops like canola and soybean. Metabolic engineering of high biomass crops like energy cane for the production of oil is a promising strategy to improve the oil yield of traditional oilseed crops. Energy cane can be converted into an oil cane by co-expression of 3 lipid genes through a complex metabolic engineering approach. Figure 1 shows a typical engineered metabolic pathway for hypercumulation of lipids. In this, the first 2 steps is to increase the fatty acid synthesis and triacylglycerol tag molecule, which is the high storage lipid molecule. Third one is to optimize its storage and finally preventing that tag from hydrolysis. So involvement of these 3 lipid genes make this metabolic engineering challenging. Comparison at single gene level would be simple but not conclusive because each variant needs to be evaluated in context of at least 2 other lipid genes. Therefore, using constitutive promoters expressing all the genes continuously may require a large number of transgenic lines and samples to be analyzed to find the few that are informative. In this scenario, inducible expression system in which gene of interest is only expressed in the presence of a specific inducer is a promising strategy to accelerate the design, test, build, and learn TPTL cycles. So for this, the objective of our study is to explore if we can accelerate this TPTL cycle for hypercumulation of lipids in energy cane by using a inducible transportation system. Our hypothesis was that using a heat shock inducible promoter for one or two lipid genes while constitutive promoter for all other genes will facilitate the variant comparison. For this, figure 2 shows we transformed the energy cane calli using a biological gene transfer in which with lipid genes in which the candidate genes under the control of HSP promoter. Results. Our results showed that energy cane can tolerate the induction of lipid genes with heat shock promoter with multiple cycles of heat treatment at 40 centigrade without necrosis. Figure 3a, this graph shows a time course experiment of heat treatment that upregulates the gene expression of two important lipid genes, DGAT and WRNKL1, from 96-fold to 150-fold. And subsequently, that led to increased oil production from 5-fold to 16-fold in stem and leaf tissues, just within 4 days of gene activation. Conclusion. So this strategy has a great potential to accelerate the TPTL cycle for identification of superior lipogenic factors, variants in energy cane for biodiesel production. Thank you.
All right. Thank you so much. Great job. Okay, so finally, we have Swati Shrestha. And go ahead whenever you're ready, Swati.
Thank you, Jordan. I'm excited to share my research with you all. Either as vegetable, soup, or pie, squash and pumpkin always find way to our home and heart. However, profitable production of squash and pumpkin in the grower's field is threatened by the infection of a virus named papaya ring spot virus, or PRSV, because popular squash varieties like butterbush are susceptible to the virus. Thus, there's a need to develop PRSV-resistant squash lines in the future breeding program. Luckily, natural source of resistance to PRSV has been reported in a local squash landrace from Nigeria called Nigerian local, and many breeding programs seek to transfer this resistance into commercial lines. Unfortunately, genes and QTL associated with resistance remain unknown, making the breeding program tedious and less effective. Now, many of you here might know what a gene is. Let me tell you what a QTL is. In plants, numerous genes with minor effects control most of the agronomically important traits, and the region of those genes is known as QTL, or quantitative trait loci. Through my research, I have been trying to identify QTL associated with PRSV resistance in squash by using a whole genome sequencing-based bulk segregant analysis technique called QTL-seq. In fact, by using this method, I have already identified QTL associated with PRSV resistance in squash in chromosome 9, as can be seen in the bottom right figure of your screen. After QTL identification, I have also developed molecular markers tightly linked to PRSV resistance from this QTL region. Now, what's the implication of all this? Let me explain this with an example. If we consider the genome of a plant as a university, then various departments can be considered as QTLs controlling different traits. Let's say you want to put up a play by Shakespeare. You would choose students from drama department instead of IT department in order to get the best performance. Likewise, plant breeders and geneticists should now focus on QTLs and markers identified through my research for the rapid selection of PRSV-resistant individuals in each generation, thus accelerating future breeding programs through marker-assisted selection. Thank you.
All right. Great job, Swati. So I'll go ahead and I'll stop sharing my screen here. Just want to say thank you to all of our participants. Let's give all of our presenters a virtual round of applause. Thank you so much, guys. You all did a great job, and you each taught us a lot about each one of your areas of research. So from here, we can open up the session to a few minutes of Q&A. So if you have any questions that you haven't submitted quite yet, please feel free to do so now. So I'm going to open up the Q&A and I'll see what happens. So here's a question for Julian. Do you know the ploidy level of the varieties that you tested, and is there a different response related to ploidy?
Yeah, actually, I love that question because it really kind of follows the path of my entire PhD. So that's actually what I'm working on now. Begonia is a mega diverse genus. It's over 1,900 identified species at this point, and new ones are being released every month. Accompanying that diversity, there's also a huge diversity in ploidy level and in genome size, both of which can have an effect on the abiotic resistance or tolerance, if you will. So have I done it yet for any of the 4 varieties? I've done it, but I haven't been able to analyze it yet. So short answer, I can't tell you. But based off the parents, for example, of FB059, I think right now we believe it to be a tetraploid, but again, not definitive yet on that. So thank you.
All right, thank you. So I've got another question here. This one's for Anju. Anju, can you talk a little bit about the different covariates and challenges you ran into in your HTP data collection with both the UAV and the ground sampling that you guys did?
Hey, Jordan, can you hear me?
Yep, loud and clear.
Can you please repeat the question?
So can you talk a little bit about the different covariates or just general challenges that you ran into when you were collecting UAV-based data, and as well as that ground truth data that you took? And were there any problems in finding correlations there?
Okay, so first of all, flying a drone or unmanned aerial vehicle, it's sometimes challenging. It depends on weather and some other things. Like if it's cloudy, you cannot fly the drone. So we need full sunlight, first of all. So it's more about flying part. And for data part, when we get the data, though you see, I'm the only person who was flying the drone, taking the all spaces from other, lab mates or labor from there. The thing is, it takes technical skill to process the data in different software to generate vegetation indices. And when we analyze the data, as I showed you in Pearson correlation, we really found very good result for that. And one of our paper already published, we found that applying high throughput data, we can even replace manual harvest accumulation by up to 50% or even 70% in some cases. Even still will get the similar prediction efficacy. So I would say it's really good for our breeding program, especially for perennial alfalfa. You know that it's very tiring and tedious when we collect data each month. Generally, we do each month, and we need lots of laborers, not only for harvesting, but also for after processing things, drying, taking wet, and data input. Lots of things are there. Yeah, thank you.
Thank you. So I've got a question here for Swati. Which methods did you use for QTL discovery for the squash resistance?
Oh, thank you for the question, Jordan. So basically, we used a whole genome sequencing based bulk segregated analysis technique. So in that technique, what happens is you collect 10 of the resistant plant samples and 10 of the susceptible plant samples, and then you send those bulk population for sequencing. And after getting the sequencing results, you look at those 2 bulk samples, genomic region, and then look at the SNPs associated with them and try to locate the SNPs and then the QTL. I saw a question in chat as well. I think that's related to me. So since I'm talking, I will answer that question as well. The question is, do you think QTL still works because we have developed CRISPR-Cas9 technology to modify the gene? So basically, I think for my crop, which is squash, yes, because a lot has been done in other crops like rice and wheat, but for squash, there hasn't been a lot. And for the virus disease and squash that I am working on, we just have basic inheritance studies. So we still don't know what gene is working on or what QTL is working on for the resistance. So for my crop, yes. But also I would like to add that even for CRISPR and other modern techniques, we need to locate and identify the gene, and the basic step for that is QTL identification. So everything starts with that. And in my case, the QTL identification is like based on whole genome sequencing. So we can locate the SNPs very accurately, and then that's the step. And the next step is we are thinking of maybe using some like genome editing technologies in our lab as well, if we have like available resources in the future. So that's all. Thank you.
All right, thank you. Good answer. Okay, here's one I have for Kasim. So it says you showed that you guys use the gene gun for transformations. Does that involve using gold nanoparticles, and how efficient is this transformation method in making successful transgene deliveries?
Thank you. Yeah, we used gold particle to bombard, and it is quite successful compared to other transformation method like bacterium.
Okay, yeah. All right, sounds good. Here's one I've got for Kaur. What are some of the challenges of scientifically improving taste, which can be a very subjective thing in tomato?
Thank you for the question. As simple as a slide looked, it is nowhere close to when we work on flavor. As I said, like it's complex in terms of phenotyping and in terms of money and everything. So with the subjective nature, when we do, so our lab does taste panels for other studies. It's not part of my project yet. So we do taste panels and all, but it's dependent on season. It dependent on like multiple factors that I didn't, explain to just for the sake of simplicity here. So all in all, in a nutshell, it's very complex and there are multiple pathways. So that's why our lab is only focusing on flavor for so many years. And a lot has been done, but we are not there yet. So yeah, I don't know if that answered the question.
Yeah, I think so. Definitely. Let's see. I think we may have a couple more questions. So here's another one for Julian. It says, so you mentioned that you're taking a kind of a suite of different morphological traits and morphological data. Are you using any high throughput phenotyping methods for that? And if so, could you kind of elucidate those a little bit?
I'm not doing that at this point because they're really, as far as research goes, there's not too much, not as much money in ornamentals compared to, your fruiting crops or anything like that. So stuff like that does cost quite a bit of money, and begonia isn't quite at the developmental stage where I think we can actually successfully apply it. Yeah, I guess you could say I'm the first in a line of many PhD students who will be working on this project. So I'm setting the foundation, if you will.
That's good. That means that everybody will be citing you first.
Yeah, hopefully.
Yeah. Okay, here's another one. This one's for Lushan. It reads, what other possible species do you think your blueberry research may be expanded to in the future? I know you mentioned that it could be expanded going forward. So what are some of those areas you think that could expand to?
That's a great question. So considering the disease that I'm working on, anthracnose, it is caused by Colletotrichum species. So, so far it has a very broad host range, including tomatoes, strawberries, potatoes, and eggplants. So these are the potential species where we can probably test the identified genomic regions. And there should be some more species that I'm not very, that I have to explore yet. But again, so far my priority being blueberries. I'm just trying to focus more on that. But yes, I mean, all the species that does have, that is affected with anthracnose, with Colletotrichum species, we can possibly test the potential.
Okay, here's a quick question. Do you ever get tired of eating all the blueberries that you guys have in that
Yes,
lab?
I do.
Oh yeah, I'm
Yes,
sure. All right, here's a question. This is not pointed to anyone specifically, but I think Swati, this is right up your area, and if anyone else has an answer to it as well. So it's, what are the challenges in identifying QTLs and genes?
I think this question is not directed to only me. I know Gurleen's research and even Lucian's research kind of is similar. Yeah, I can give my perspective about it. So that's a good question. Like, what are the challenges for identifying genes and QTLs? Basically, I think that for QTLs, there are a plethora of methods that can be used for identifying QTLs, and each method has its own pros and cons. I can talk about my research, my method that I have been working on since like last 2 years. So in my method is really quick. What we do is we just have developed F2 population, take 10 resistant and susceptible cell number sequencing, get back the results and just analyze it bioinformatically. So in my case, we get like big QTL regions, like they can be like 1 million base pairs. And from that QTL region, if you have to narrow down and find the genes, that gets really difficult because there are like so many genes in that region, and then to specifically find the gene that is related with my disease resistance. So I think that's probably one of the big challenges. But then again, we have like so many softwares available, some of them really accurate. Like the one that I use is called SNP Effect software, which can really pick out the SNPs from those regions that have very high impact. And then that has very high accuracy. So in my case, that's the thing, like big, like narrowing down the genes from those big regions is kind of a big challenge. But then also, even though we have like found out many genes, doing some expression analysis, testing the markers kind of helps to narrow that down. So that's my experience.
Okay, thank you, Swati.
I might jump in, just one quick comment. So I agree with everything with Swati just mentioned. I think my work is a step behind because basically we are trying, so as I mentioned, I'm working with 3 diseases like in this presentation, and Downy mildew is the most important worldwide for lettuce. And the idea is to try to find accessions with multiple disease resistance, because in a breeding program, like it takes forever, 8-10 years to develop a resistant cultivar. So QTL analysis helps to, decrease this time. But in my perspective here from my research, we try to use some accessions or that is not cultivars yet to be tested with other pathogens. And then if we can combine in one single accession multiple disease resistance, we can breed elite breeding lines to incorporate in the breeding program. So we are just trying to find as many tools as we can to improve as faster as we can the plants.
I'd like to add to what Liz said, if we have some time. Yeah, so sometimes, especially when we're working with certain pathogens, the resistance, it's likely to break down at higher temperature, and under lower temperature, it's still the species could be resistant. So sometimes when we do identify QTLs and use it for breeding purposes, but eventually at certain different temperatures, it might still not work. So it's not exactly a challenge in identifying QTL, but it is a challenge after, after applying the QTL that was identified, if that adds up.
Yeah.
Thank you.
If I could jump in just one more time. So as like the theme of this symposium is climate change, and it's believed that outbreaks of diseases, in our case that we work with diseases, are being more frequent. So doing this kind of analysis is very important, because like even though we breed for many years or shorter years a variety that is resistant, the outbreaks are happening. So the breeding is a continuous work. So yeah.
Yeah, truly so. It's definitely continuous work. It's always going. Yeah, thanks so much, guys. I think we have enough time for maybe one more question. We have a question for Kar. What are the characteristics of good flavor?
Okay, so as I mentioned in my slide, sugars, acids, and volatiles, I said 400, but our lab narrowed it down to like 30, 33 that we usually test the varieties for. So when you ask for what are I can count like a few only. I can't do that, but we have like a profile, a set of volatiles that's why it's so complex because of the interactions and overlapping pathways and all. So overall, you can say sugars, acids, and a bunch of volatiles in short. Thank you.
Thank you. Yeah, I've had many different tomatoes and they seem to all have very different flavors. Sometimes you get a tomato that tastes bland like nothing, and sometimes you just get that super juicy, sweet, acidic tomato, and it's really, really good. So yeah, your work is much appreciated in making those tasty tomatoes.
Thank you so much.
All right, guys, so we're getting close up to lunch here, so I think I will go ahead and we'll wrap up the Q&A. You guys all did a very good job, great presentations, great Q&A sessions, really good questions, everybody. So we'll go ahead and we'll do lunch, and so that will be that'll go up to 12:55, at which point we will have our next speaker, and I will be handing over the reins then. In the meantime, I will be tallying up your guys' points from our 3 judges. So if those 3 judges want to get those score sheets back to me ASAP, that would be great. And then during the closing remarks and award ceremony, I'll go ahead and I will announce our 3rd, 2nd, and 1st place winners. So if we can just do one more round of applause for all our presenters today. Thank you guys so much. You guys did great. All right, and like I said, lunch will be up until 12:55.
Hello there, organizers.
Hi, Dr. Morris.
Hello. So, you want to test sharing my screen?
Yeah, I think that's a good idea.
Hi. Okay, let's see. Okay. It says host disabled participant screen sharing.
I just made you a co-host, so you should be able to share now.
Okay, let's give that a shot. Yes. Okay, presentation mode. Can you see my screen?
Yes, sir. Perfect.
Okay. And if you wouldn't mind if some of the organizers would stay with their video on, it's just a lot nicer to give a talk when you can see some humans reacting.
Sure, no problem.
Sure.
So let's just wait a bit, and then I'll make a short introduction for you, sir, and we'll get it on.
Sounds good. How's the meeting been going so far?
It's been super interesting. A lot of good presentations. The student talks were great, I think. It's going very good.
I love these student symposiums. They're like the best way to actually meet people and talk science.
I agree. I think we should wait until 12:55, as we said, right?
So let's see. Okay, so 12:55.
2 minutes.
So what crops does everybody work on? Let's see. Swati, what do you work on?
I'm working with squash, so basically like developing molecular markers for virus resistance in squash.
Nice.
That's like my general description of the project, but papaya ring spot virus and zucchini yellow mosaic virus resistance.
Nice. Manuel?
Yeah, I'm working with vanilla. Yes, sir.
Vanilla?
Yes, sir.
Orchids, right?
It's an
Yeah.
orchid, yes, sir. I love orchids.
Yeah, there's a charismatic plant, right?
Yes, it is.
Nice. Joshua?
I started out working with passion fruit, but I also picked up working with vanilla as well. So me and Manuel are in the same lab.
Okay. Wow, it's really different. You know, I was at Kansas State for 6.5 years, and it's like wheat, and then like one sorghum person and one soybean person. A lot more diversity at Florida.
Tropics.
Yeah.
We're actually getting pretty worried with this cold spell coming through. I think we're almost like the world's coming to an end in a little bit with the freezing temperature. But we'll survive.
Okay, I guess we're on time. I guess we can. Hey everybody, welcome back. Hi, Dr. Morris. We're very happy to have you join us in our 6th annual Plant Science Symposium. I would like to start by giving a brief introduction of Dr. Morris. Dr. Geoffrey Morris is an associate professor of crop quantitative genomics at Colorado State University. His research group develops and implements new approaches to understand and improve crop adaptation towards climate change resilience. His methodologies include mapping of genes that underlie climate adaptation and develop new approaches to better understand this process. His focal crop is sorghum, and he and his team work directly with breeding programs in developing countries such as Senegal, Niger, Haiti, amongst others. He's passionate about international research and education, focusing on soil, water, crop management practices, nutrition, and stress tolerance for improving crops productivity. In the US, he also works to facilitate the development of better adapt sorghum varieties in Kansas, Texas, and South Carolina. So with no further ado, please, Dr. Morris, the floor is yours.
Thanks so much. I am really glad to be here. And I just wanted to start giving a little overview of what I'm going to do today. Since I'm at the very end, and you've probably heard a lot of science already, and I'm really passionate about introducing students to the opportunities to use the scientific method. I'm going to do a talk that's a little different today. I prepared a special talk just for this symposium. I'm going to show you how you can use an applied scientific method to help you in your career. And I will talk about my own lab's research, but I'll be kind of using it really to give illustrations or examples of how you can use an applied scientific method. So I'm going to start by talking about why. Why do you do what you want to do? So how to develop your vision and mission, and to convince you, yeah, you know, you can think about trying to change the world, make a better place if you want to. And if you really get the goal setting right, you can really do that. The next step is really going to be that goal setting of what you actually do to understand your next user's goals and to set your goals to be able to help your next users. And then the how do you do that? How do you achieve your goals? How do you help your next user's goals? I'm arguing it's really through the scientific method. The amazing thing about hypothesis testing is you can start with the wrong answer, and hypothesis testing will get you to the right answer to achieve your goals and then eventually see your vision come to fruition. So I want to start by saying, you know, some scientists really have saved the world. You know, if you're willing to round up, Norman Borlaug credited with you know, basically 1 billion people not going hungry because of the work that he and others did. A couple kind of big picture lessons from the successes in the past. It wasn't hard, it wasn't easy, and it wasn't quick, right? And it wasn't done alone. You know, it's important to idolize people like Borlaug who had a great impact, but it's also important to remember there were so many people who were involved in making that success. He was just one. I just brought up some key names, but of course there's dozens of people who devoted their careers to making this success happen in, for instance, the Green Revolution. So I'm going to start by telling you my vision. This is what motivates me. I have the vision that science could provide food security and economic prosperity for the world's drought-prone regions. This is what I think of when the work gets hard, when I get frustrated, when I get tired. I come back to this idea that this could happen. Why do I focus on this? It's because There's about 2 billion people in these drought-prone regions, and about 1 billion of them are in pretty abject poverty. And we also know that climate change impacts are disproportionately already affecting these people, and are expected to do more of that in the future. And when I say this, you might feel reasonably enough like, isn't this too big for one person, right? And of course, yes, absolutely, you're right. It is too big for one person. But I want to say it's okay to start by saying what you care about, right? And what about specific responsibilities, right? Who's going to do this? What am I going to do? It's important to take on responsibility, but I'd say first, it's better just to start with saying what it is that motivates you, right? Let people know about that. See if you have a shared vision. So the mission of my program is to understand and improve crop adaptation to drought-prone environments. And so I do focus on this crop, sorghum. It's a food and forage and feed crop around the world in these drought-prone areas. And so how is that mission different from the vision? The difference is I am taking responsibility for this mission. I hope that I'll meet some of you again in 10 years or 20 years, and you'll say, you told me you were going to understand and improve crop adaptation to drought-prone environments. What have you done? What have you done for that, Jeff Morris? And I work every day to make sure that I have something to tell you in 5 or 10 or 20 years when you ask me that. So I'm taking responsibility for that. You might wonder, where are the specific goals, right? And those are coming, but we want to think through our motivation kind of step by step. So the particular story I'm going to tell you about today, coming out of my lab, is the evolutionary rescue of sorghum, which is a climate resilient crop after a really terrible aphid outbreak that occurred in the Americas in the last decade. So this paper is coming out in a couple of weeks in Science Advances. And the reason this was of interest, for a broader interdisciplinary audience is a couple of things. We showed that half a century of shared work that was necessary for this rescue of sorghum to happen after the outbreak. We traced that history. And we also showed how you could use an understanding of that evolutionary rescue in order to rapidly develop new technology that will hopefully prevent some future outbreaks. Okay, so this outbreak was of the sugarcane aphid on sorghum, and so there's a biotype shift similar to what we're seeing now in COVID with the new variants that led to a really widespread outbreak in the Americas starting in around 2013. And so in the commercial sorghum agriculture, what that led to was a massive increase of insecticide use, and so that really reduced profitability for the growers and of course reduced the sustainability of the system as well. And that led to a very rapid decline of acreage for this climate resilient crop. It meant that the crop was getting substituted for much more drought sensitive crops in many cases. In Haiti, it wasn't just an economic issue, it was a food security issue. Haiti, the poorest country in our hemisphere. It's in the Caribbean. It has some humid parts, but a lot of the agricultural regions are actually quite dry in the rain shadow of the mountains. And so sorghum is the most important, really climate resilient, locally produced staple cereal in Haiti, along with rice and maize. It's an important part of the diet. And from 2015 to 2017, the sorghum landrace has almost disappeared. This is a sorghum field that got completely wiped out in Haiti that I visited. This is one of the few plants I could find that was at all surviving. Most of the plants died. The farmers replanted with corn. So they had a corn field, but that corn, as we know, is more susceptible to drought. And so this is basically what was left of the sorghum in a lot of places. And so there was a massive reduction in 2 years of the sorghum production, basically down by over 100,000 tons in this small country. So what happens when there's a huge change in environment like this? So in the ecology and evolution literature, they have this concept called evolutionary rescue. So you have this rapid environmental change, and there's a couple possibilities afterwards. One is, of course, extinction. The population level could get so low that it gets past a critical point where it cannot recover. And the other possibility is evolutionary rescue. And that is really just genetic adaptation due either to new mutation or to standing variation, polymorphism that's already there somewhere in the population. And this is, thankfully, what occurred in the situation in Haiti. There's a wonderful, wonderful breeding program, very new, by the University of Quisqueya. And they had just started a new breeding program when the aphid hit, and they were applying very intense selection for the aphid, and with no insecticide on a very diverse program that they had just built. And almost the entire program was wiped out. There was just a handful of survivors, but they were able to use those survivors, intercross them, rebuild a whole new breeding program, and very rapidly, incredibly rapidly, develop a new SCA resistant variety. And because of that, the sorghum production rebounded very quickly. A really incredible success. And that's really what we wanted to study. Sorghum was rescued in the Americas in just a matter of a few years. On the commercial side, the commercial sorghum side, there was existing hybrids. A few of them were found to be at least somewhat resistant, and those were released. So our question was really, how did this rescue happen, and how can we leverage knowledge on this evolutionary rescue in order to do a couple of things? One is to accelerate the development of new resistant varieties, because only a tiny number of varieties kind of made it through that bottleneck, right? So there need to be, they weren't the best varieties in other ways. So there needs to be more work on developing new resistant varieties. And also, how do we use this information to prepare for the next aphid outbreak or future environmental change? Okay, so I want to shift gears now and talk a little bit more about deciding what you're going to do. And I told you my mission already, and as was said in the introduction, I'm not a plant breeder, right? So how in the world can I achieve my mission if I'm a geneticist and not a breeder? And then the way I do that, right, is working with breeding programs. We work with breeding programs around the world. These are the public programs we work with. We also work with a number of commercial programs in the US. And we have to make sure our discoveries are designed to make it into those breeding programs, or else the program really doesn't have much of a direct impact. The way we do that is through these trait technology packages. And so typically, if the breeding program we're working with doesn't have the adaptive trait allele, then we will make a trait donor. So taking that allele and introgressing it into a suitable donor line for that breeding program. We'll make a trait marker, and that'll be low cost and will be outsourced, easy to use. So these programs, these breeding programs, don't all have to become experts in physiology or entomology or whatever the adaptive trait is related to. And then we'll make what I call a trait map. And so this is like the scientific models of like, how does this trait work? How do you need to use it in the breeding program? So to get to your mission, right, you need to figure out who your next users are and what their goals are. And so this was my mission. And so I spent a lot of time talking with the breeders in Haiti and the breeders in the US trying to understand what they needed. And so on the Haitian side, they had the resistance, but they're very worried about the next biotype shift, the type of thing we're seeing with COVID now, right? One shift, one variant after the other. So they want to develop varieties with more than one source of resistance. On the US side, their biggest challenge is they need to convert these great hybrids they already had in the pipeline into resistant hybrids. Oops. Okay, and so before we made our goals, we first did this extra step of understanding our next users' roadblocks. So what do I mean by that? Like, where in their goals are they getting stuck? So in Haiti, the issue was they can't select for a new gene when the old gene still works, right? They could do the phenotypic selection very effectively. They showed that, but they don't know whether they're just reinventing the wheel every time, right? With the existing gene. On the US side, it's a little different. The aphid pressure is really bad on farms, but it's very patchy. So phenotypic selection is very inconsistent and very expensive. So it's very difficult to convert the varieties, their hybrids with phenotypic selection. So our research goal now, with that understanding in mind, was to identify a cheap, accurate, and global SEA-resistant marker that these breeding programs could use to select for, or depending on what they're doing, select against known resistance. So we developed and tested an SEA marker, and I'm not going to tell you how we did it quite yet. I'll come later. But we developed a CAS marker because these are low cost and outsourced, and these small breeding programs like in Haiti or mom and pop seed companies in the US don't need to develop a marker lab. And we tested these in an independent breeding population, or really just family that we had developed crossing US and Senegalese lines. And we used a greenhouse assay to test for the SEA response. And so here's one of the resistant progeny on the left, and the susceptible parent getting completely demolished by the aphid. And so, we genotyped these lines that we're testing, so we could classify them into homozygous for the susceptible associated allele, homozygous for the resistance associated allele. And you can see there's an almost perfect correspondence then between the phenotypes, which are there in this histogram on the x-axis, and the prediction based on the marker. So that gave us some confidence we were on the right track. But remember, our goal was to make a marker that was effective globally across different public programs and different private programs. And if you know the history of marker-assisted selection, there has been incredible success developing markers for individual programs, and a lot of disappointment developing markers, especially in the public sector, that were meant to be used broadly, right? In most cases, they didn't end up working for a lot of traits. So we also tested them in these markers in 4 commercial seed company programs, US seed companies, and 3 US public programs, which are kept anonymous since there's confidential information involved. And overall, we saw usually around an 80% match between the phenotype and the as determined by the breeder's own ratings to the markers. And so there was mismatches, which are important, right? Because this is what has doomed some of these efforts to make global markers before. So we know one issue is definitely that there's inconsistencies in the public germplasm, because we used public germplasm as for a lot of these checks. And we know that now over the last, 10 or 30 years since some of these public lines have been developed, things have gotten mixed up across the community, and that's always comes up whenever whenever there's new marker work. You always reveal what's been going on with the germplasm. We know there's phenotyping issues. That's why these programs, came to us or were willing to partner with us, because the SCA ratings in the field are very inconsistent. The other possibility is that there's complexity to the haplotypes. maybe it's the same gene involved, but the haplotype that we're tagging with the marker is different, or there can be other genes. So there's a lot of potential hypotheses to explain what's going on, but overall, we were able to show that these markers were predictive. And because of that, then we were able to meet our next user's goals. So now this marker, which costs $2 all in to get per sample to get the genotype, it's being used by public programs in Haiti and the US, and now we're working with a program in South America. And one of the US seed companies, for instance, in the first year has ran more than 10,000 samples with this markers. So we're at a point now where we can say, yes, we've helped our next user achieve this goal, and pretty quickly too. So what I haven't told you, but maybe you're wondering, is how did we develop the SEA resistance marker? And how did we know, or why did we suspect the SEA marker would work globally? And that's kind of the fun part of this study, because it really brings in a lot of evolutionary and population genetics foundations into this marker development work. And the whole thing was really done with this goal-directed, hypothesis-driven research framework that we're using. That was one of the reasons why we were able to do this so fast. So how does hypothesis testing work? How does it get you to the right answer? So, I told you the research goal, but I didn't tell you about the hypothesis underneath it. This is the research goal I mentioned before. And so the first key point on this hypothesis testing issue is, that I'd like to convince you of, is that underneath every goal, whether you say it or not, underneath every goal, there is a hypothesis. And you can choose to assume the hypothesis, basically, and not state it, but it's still there. So why am I saying that? It's because, if you ask yourself this question, why do I think this goal is realistic? Why do I think this thing can be done? You have to think that there's some reality out there in the world that makes it so that goal is realistic. Something has to be true for the goal to be realistic. And that's what the hypothesis is. That's how you can find the hypothesis, is ask yourself, what needs to be true for this goal to be possible? That's your hypothesis. So for us, when we were developing the hypotheses, we did what everyone does, what you're supposed to do, dive into the literature to try to understand what was already known. In the aphid resistance, sugarcane aphid resistance case, there was one Chinese group that several years before had identified aphid resistant line, sugarcane aphid resistant line here, HN16, had crossed it with a US susceptible, and had mapped this RMES1 locus on chromosome 6. And so that's interesting, right? And the natural first question is, does RMES1 exist in other germplasm? Might it underlie resistance that we're seeing elsewhere in the world? But unfortunately, this HN16, there's nothing known about the origin, not available outside China. It's not easy to exchange germplasm with China. So on its own, it could be kind of a dead end. But this is where the evolutionary and population genomics kind of came in and unlocked the power of the work that the Chinese group had done. So, so we have this research goal, and then we need to kind of articulate the hypothesis, or a hypothesis that would make this research goal be tractable, right? So one such hypothesis is that SCA resistance in Haiti and in the US is due to RMES1. If that was true, right, then we should be able to track down alleles in the Americas that tag that RMES1 variation and make the markers. So the question, though, is how do we test this hypothesis? We don't want to just assume this and rush forward on a lot of marker development without knowing whether this underlying premise is true. Because that's why, one of the reasons maybe why some of the public sector marker development in the past has underperformed. So in this case, we could leverage this this set of concepts from evolutionary genomics, and that is that selection reduces genomic diversity, specifically in the loci under selection, for instance, by the breeders. So we did high density genotyping of the Haitian breeding program, several hundred lines that were developed after the sugarcane aphid selection, and we compared that to the genome-wide diversity for global sorghum. So in this plot, on the x-axis, we have the 10 sorghum chromosomes. Each position on the x-axis is a position in the sorghum genome. And then on the y-axis, we have a measure of nucleotide diversity, SNP diversity, so pi. So higher is more diversity, lower is less diversity. And you can see that amazingly, the Haitian breeding program still has a ton of diversity, only about a 50% reduction in diversity relative to the entire species, which is pretty amazing. And that was really because of the design of the breeding program to be very diverse. But if you look closely, you can see that there's regions in the genome that have lost a huge amount of diversity. And that is, based on theory, that's where we think the breeder's selection was applied. And so we've noted a few genes where we thought a priori, based on the breeding targets for this program, there was probably selection, for instance, against tannins or for stagrine. And so these signatures give us a way to identify potentially the signatures of the aphid adaptation. So there's two signatures. There's low diversity, and that's what I'm showing here. And then there's high divergence, which is the other signature of positive selection. And that's what we looked at next. So notably, RMES1 is at one of those spots where there's very low diversity. So can we fine tune this with the signature of divergence? So in this case, we were using fixation index, a very old evolutionary genetics concept going back to Sewell Wright 100 years ago. And it's just this classic measure of divergence. So we calculated FST for Haiti versus the global panel for each SNP across the genome, hundreds of thousands of SNPs. And under the hypothesis that RMES1 underlies SCA resistance in Haiti, then there should be significant FST outliers at RMES1. And so I'm going to show you how it turned out. Again, we have the 10 sorghum chromosomes on the x-axis. Now on the y-axis, what we have is the significance of the FST. So when this value is high, that means you have high divergence, which is what we would expect under selection. Okay, and so this is exactly what we expect. This is exactly what we would expect under the RMES1 hypothesis. RMES1 is found at the genome-wide peak for selection. It appears to be the locus under greatest selection in the Haitian breeding program. And that's true when we zoom in as well. This in red is the original interval identified with linkage mapping by the group in China. And here is the peak FST, highest divergence, highest FST SNPs right here. Okay, so, but we as a program that supports breeding programs, we need to understand this in the context of the breeding programs as they exist if we're going to make a marker that serves the needs across many public programs. So I spent a lot of time digging through like old pedigree records from like the 60s and 70s and 80s to understand where the germplasm people were using, where it came from. And was there a connection between the work, the aphid resistance in Haiti and the aphid resistance in the US and elsewhere? And so it turned out there was something that looked like a connection. This TX 2783, this was the major public SCA resistance line that was identified, and others with this SC110 in the pedigree. And it turned out that had the same progenitor as a West African SCA resistant variety, which was itself a progenitor of the Haitian breeding population. So we had a connection. In purple, we have the SAA resistant parents, and in gray, the susceptible. And so then the hypothesis is that based on this pedigree, there was a shared resistance allele, which could be the basis of a shared marker, a global marker. Let's see. Okay, so we also could make observations about the genome-wide kind of genetic similarity and structure of these populations. And we saw that the Haitian breeding program was basically nested within Ethiopian diversity primarily, whether we look at it with a principal components analysis on the SNP diversity or a neighbor joining tree. It showed pretty clearly that the vast majority of the diversity represented in the Haitian breeding program was diversity that came from Ethiopia. The breeding program knew that they used a lot of Ethiopian diversity, but they also used other global diversity. And so with this information, then we can develop a hypothesis really rigorously on the origin of RMES1. One thing to point out, to remember, scientific method is really about excluding hypotheses, right? It's not about proving hypotheses, it's about excluding them. So how do you develop hypotheses that you can exclude? One, you need to have two hypotheses, right? Because you're going to exclude at least one. So you always have two competing hypotheses. And then you take existing theory and previous observations and you make new hypotheses with that. So in this case, we use the kind of established theory in the sorghum world that STA resistance is not common, been tested many times over many decades, with the observation that the Haiti lines cluster with the Ethiopian lines. Then we can make the hypothesis that RMES1 in Haiti is a rare variant from Ethiopia. And then contrasting to that hypothesis, we can take different theory and different observations and make a competing hypothesis that RMES1 is not a rare variant from Ethiopia. Maybe it's really just a Chinese thing. Maybe it's actually spread all over Africa. We know that STA exists across Africa, so maybe there's been selection for it all over Africa. We know that the resistance was, association was originally found in a Chinese line, so maybe it doesn't have anything to do with Ethiopia. So now we have our competing hypotheses. So the next thing we need to do to exclude one of these hypotheses is to make predictions. Okay? The only way to really rigorously exclude a hypothesis, use the scientific method, is with a priori predictions. So these are the same hypotheses as before, and now we're going down to making a priori predictions about what we should see in our data if that hypothesis is true. So in the case where we, under the hypothesis that RMES1 is a rare variant from Ethiopia, then when we look at global diversity of sorghum land races, the resistance-associated SNP allele should really only be found in Ethiopia, right? By contrast, if that first hypothesis is true and the competing hypothesis, if the first hypothesis is false and the competing hypothesis is true, RMES1 is not a rare variant from Ethiopia, then there will be resistant associated SNP alleles, say, all over Africa, or not in Ethiopia, or only in China, something else, right? And we can visualize these predictions. Here I've kind of mocked visual predictions up after the fact using and modifying the real data. But under the for instance, under this second hypothesis, we would predict these resistance-associated alleles noted in red. We would, predict that they will be outside Ethiopia, right? Okay. So then, what did we see? We saw, and so now here we're looking at the real data. The susceptibility-associated allele was almost fixed in the U.S. Just a few of those resistant lines were there in the U.S. In Haiti, as we already knew, the resistant associated allele was almost fixed. That's how we found this particular SNP here, which is the associated SNP. Okay, so that makes sense. Now, what about in the rest of the world? How did it look? So if you notice, the resistant alleles are only in this one corner in Africa when we're looking at global sorghum landrace diversity. And that little corner is, of course, Ethiopia. Right? So this allele is really only found in Ethiopia. And so then that allows us to exclude one hypothesis and at least tentatively accept another. And that is that RMES1 is globally rare, but locally common variant from Ethiopia. And so just to kind of really show kind of rigorously what I mean by excluding of hypotheses using these predictions, right? So just replotting what you saw already before. So our observations matched the prediction under this first hypothesis and definitely did not match the hypothesis. Did not match the prediction under the second hypothesis. And so that means we can exclude the second hypothesis, right? Now, one thing's important to say, we didn't prove the first hypothesis, right? Because you can't really prove anything conclusively with the scientific method. What you can do is exclude hypotheses and see what hypothesis keeps surviving, experiment after experiment, prediction after prediction, right? And so we haven't cloned this gene yet, right? And so there's still the possibility that we will, when this gene gets cloned, we'll find out that there's something else going on. And so that this hypothesis was wrong, but this hypothesis lives to fight another day, and we'll keep testing it as rigorously as we can. Okay, so overall, what did we find in this story? This rapid evolutionary rescue, incredibly fast, just a couple years. It seemed really fast, but it took 50 years or more of global germplasm and knowledge sharing. So going back to that point, it wasn't quick or easy as it looked, and nobody did it alone. I'll just take you a whirlwind tour of the world for the path of RMES1. So, and then it'll be color coded by basically kind of like the decade that it happened in. So we know we have pretty good evidence that the allele originated from Ethiopia, right? Hundreds of years ago, thousands of years ago, we don't know when, but we know that in the 50s and then in the 70s, the Ethiopian gene bank shared it with the global gene banks, shared this one key accession, and that accession got over to African breeding programs, nothing to do with aphid resistance, just because it was a nice landrace that they wanted to use for breeding. And then through the 80s, 90s, and 2000s, the allele got shared around by breeding programs in West Africa and then back over to the US. And then in the 2000s, the resistance sources in the US germplasm were identified, but not in the US, because the aphid wasn't an issue in the US at that time. It was actually in Southern Africa, that the aphid resistance was actually found, and that information was sent back to the US, and as well as being used in Southern Africa. Then there was the discovery of RMES1 in China. And then finally, most recently, these collaborations between breeders and geneticists in US and Haiti and Senegal that allowed us to put this all together. So not quick or easy, and definitely nobody doing this alone, but the breeders, the gene bank scientists, the entomologists that were working on this really did save sorghum through this half century of work. I love this story because it really highlights the connections between all of us around the world, right? We have this one allele that connects African smallholder farmers with Haitian smallholder farmers and US commercial growers. And obviously it also connects those of us who consume this as a grain or consume the animal products that are fed on the sorghum grain. We're all connected, and the development of this market really brings that home. Okay, so what have I done today in this talk? I've try to give you a snapshot of this logic chain that we use in my group. I teach my students. I set this training material up and the framework up, but really what I was doing, I was reverse engineering how the best breeders, the best the best crop scientists that I worked with, how they thought. Okay, so Gael Pressoir, the breeder in Haiti, is an incredible visionary who sets goals and makes it happen. So like this framework is basically a reverse engineered version of how somebody like him thinks, or the wonderful wheat breeder at Kansas State University that I worked with, or many breeders that I met in industry. So breaking it down from the top to the bottom, why do we do it this way? And what's the what's the rationale for this? We need to first understand our own motivation, right? If we're going to be successful, if we're going to be a good collaborator. And so this question of why you do what you want to do, what I want to encourage you to do is take the time that you need to understand and to explain your own motivations. That's what I'm suggesting. And the flip side of that is don't rush into trying to define specific objectives. I started my lab, everything was GWAS, right? Because that's what I know how to do. That was my hammer and I was looking for the nail. Now I take much more time to understand why I'm doing what I'm doing before I rush into picking a particular approach and then trying to fit it in somehow. What you do. That is the key, is learning your next user's perspective. This is something I definitely learned from my industry collaborators, right? They're so good at that in industry and being focused on what their customer wants. We can definitely use more of that thinking in the public sector. So learn what your next user's goals are and why they're trying to understand why they're getting stuck. Then you can design your research to address the roadblocks, right? That same idea of just not rushing into exactly what you're going to do. And then the last piece, how you're going to get there. What I'm encouraging is really committing to the scientific method, taking that time to discuss your hypotheses with your colleagues, write down your hypotheses and predictions. It is like really, really fun. It makes science so much more rewarding. And it makes it more effective, I believe. And I have templates and training material that I'll share with you at the end to help you do that. So this last thing, I heard this quote first from Pearl Millet breeder in Senegal that I work with, the quote from Borlaug on that the first essential component of social justice is adequate food for all. And these are pictures from Haiti, kind of really underlying that connection. These are two pictures from the same hill. The ruins on the left, this is the fort where the rebellion really started, where Haitians overthrew slavery. On the same hill, just turning around, we're looking down on a field. This is a seed production field for the new aphid-resistant variety that the Haitian breeding program developed. So with that, I'd like to thank the members of my lab that did a lot of the work that I talked about today, and also especially thank my collaborator in Haiti, Gael Pressoir. And if you're interested to learn more about how you could use goal-directed, hypothesis-driven science, you can go to gohi.org. Thanks very much.
Thank you, Dr. Morris. I think that was an incredible presentation. It was very easy to understand all that you said, and it was, well, I want to say from my part, and I think everybody very motivating. So thank you a lot. We have here a couple of, quite a number of questions for you. So yeah. Here's one. Are there any other traits of interest that could be linked to the RMES1 marker derived from Ethiopian plants?
Yeah. Okay. So that's an interesting one. Let's see if I can answer that without skipping back to the slides. So yeah. So this was a really neat example. This question brings up how the surprising ways that actually understanding the genetics of the trait can help, because when I started talking with breeders about aphid resistance, do you need help? Some of them said, no, we got it. But, and, others were like, yeah, and actually it's really hard to phenotype. But one of the things with RMES1, it is actually like right beside one of the major flowering time genes in sorghum. And so one of the hypotheses we're testing with the Haitian program right now is they are a bit concerned that there is linkage between one of the flowering time alleles that they need or that they've in the past used and the RMES1. And so that's one thing that we're looking to. But the more we learn about the genetic basis of the traits, the more we're going to be able to actually anticipate problems of linkage drag instead of just pushing forward the breeding for 10 years and then realizing the problem afterwards. So that's actually the focus of a follow-on project that we're doing with the Haitian program, is dealing with those linkage issues. It's a good question. I see I have the Q&A up, and I see from Jesse Murray again, perceive a lack of evolution-based concepts or analysis in breeding-focused research. That's such an interesting question. I say I see sometimes like too much and also too little. So my background is evolutionary genetics. I was really excited to try to apply evolutionary genetics to concepts to breeding. And I first started really working with crops as a postdoc research assistant professor in South Carolina with Steve Kresovich. And he was like, what everybody says about crop geneticists? All they do is come in after the breeders have done all the hard work and let's just make a nice paper out of it. And that really stuck with me. And I think that perception is largely true, right? I don't think the breeders are that far off when they say that. That is mostly what geneticists in the public sector have done. And so this paper, I'm so proud of it because it's the first thing I've been able to hold up and say, here, evolutionary genomics made this tool. And this marker would not have happened because other people are working on markers. They just don't have them done. We did it faster because we were able to leverage, number one, a great breeding program. It would not have worked if the Haitian program wasn't so well designed. But we also leveraged evolutionary genomics. And so to my knowledge, this was the first example where we proved that evolutionary genomics was useful to breeding. Yeah, so I think there's a lot more evolutionary. I think there's a lot more that evolutionary geneticists could do to meet halfway. That's kind of my thought. Okay, what else?
We also have a couple of questions that are coming here through the chat. Here's one. Do the aphids carry any other disease, or is the foraging the main issue with the aphid?
Yeah, so it's like, do they carry like viruses or something like that? I've not seen anything in the literature to suggest that that's the case, but it's a great hypothesis to consider. And I do know my colleagues down the hall who work on aphid virus interactions are keeping an eye out for any evidence that hypothesis might be true.
Yeah, well, that question was also related that it should be easier to discover well, let me just read it, please. It should be easier to maintain resistance to the insect pest than fungal or viral pests. But is it easier to discover it?
Easier to maintain resistance. And you're thinking that the biotype shifts are in the aphid are slower than the, I'm blanking on the technical term for the shifts in the fungal or bacterial side, but basically you're saying the idea is that the arthropod pests evolve more slowly.
Yeah, I think
so I don't know. So they're in sorghum, there's pretty good evidence that in the 80s and like late 70s to the 80s and early 90s, basically all genetic gain stopped in sorghum because they had this green bug aphid problem. And there was, I think, like 3 or 4 major biotype shifts. So they happened really pretty quickly. So I think the idea that we might have to deal with biotype shifts, I think it's fairly likely, especially because it seems like most of the industry is using, or like most of the community is using probably one major gene. We do suspect that there's more. What was the second half of that question? Sorry, I forgot.
So was it easier to discover it?
Oh, yeah. The only reason this looked easy is because of how hard the Haitian program worked and how well designed that breeding program was. Those evolutionary genome scans, there's hundreds, thousands of papers, in crops using those evolutionary genome scans. Why haven't all those turned into markers before? It's because they weren't doing the genome scan on a basically perfectly designed breeding program for those genome scans.
We also have another question here. Can you talk a little about how you identified the SNPs associated with the resistance and developed the CASP markers?
Yeah, sure. So the main goal of this project is a project with the Gail Pressoir, the Haitian breeder, and Ed Buckler, Cornell, funded by USAID. And the main goal was to test whether genomic selection in a developing country breeding program was actually feasible. It turns out it is. We've been doing it now for several years. My lab's been helping, but the program in Haiti has been doing it for several years. So we had genotyping by sequencing data for the whole training population. So that was where the SNPs came from, was genotyping by sequencing. So fairly high density. We're not getting all the variants though, right? And so then how do we identify it? It was really just completely off the shelf genomic selection, or sorry, not genomic selection, genomic signatures of selection tools. In the world I came from, FST, the old test, nobody would use that. You'd be embarrassed to try to publish a paper with that because there's much cooler things. But I picked that on purpose because if any of you ask me, like, how does FST work? I can actually write it down. I can write down the equation. The new methods, I, even as a professor of quantitative genomics, don't understand all of them. But we just took the simplest method and we set up our hypotheses well. We built off a really great breeding program. And then we got a very clear answer. It might have gone the other way, right? We might have seen no signature of selection on RMES1. If that had occurred, we would have had very, quite conclusive evidence that it was a different gene, right? So either way it turned out, it would have been useful. It would have been interesting.
And I think we have maybe time for one last question. I have here, what was the behavior of the heterogous zygous plants for the resistance genes? Do you know
yes.
information about that?
Yeah, okay. So we did all our studies with homozygotes, in part because we had the markers, so we could find the homozygotes. The training population in Haiti they were all inbred at the point that we were studying them, but it was already known in the literature the resistance is dominant. So actually, a really nice control that maybe we should have done in that first validation that I showed you, that kind of U-shaped curve. A nice extra control would have been to make sure that the hets have the right aphid resistance reaction as expected. Actually, the reason we did that is because we didn't do that is, we needed to use, I think our entomology colleague used maybe 6 plants for the ratings. You needed multiple plants. That's actually why we couldn't use hets. But yeah, it should be dominant.
Okay, Dr. Morris, thank you again for the beautiful presentation and excellent explanations of everything. I think everything was very clear. And now with this, we conclude our presentations for the Plant Symposium. I want to thank all of the speakers also for their incredible presentations, and I would like that our attendees also thank them for logging in. It means a lot for all the Plant Science Council at the University of Florida, and it's very important, all the knowledge that we've shared here. And now, as promised, now we will reveal the winners of the Flash Talk competition. So now I would like to pass the floor to Jordan so he can tell us. Thank you.
All right, thanks so much. Great presentation. All right, everybody. So now comes the time to announce our awards. Before we get to that, though, I'd just like to thank all of our participants for their efforts in making their slides and delivering their presentations. I think everybody here really learned a lot from your guys' research. I also want to thank the judges for their participation in scoring each presentation, as well as everyone in the audience that decided to tune in and to watch those. And finally, I'd also like to just take a moment to thank Josh, our president, for really spearheading this year's symposium efforts. So without further ado, I'll go ahead and I'll announce our Flash Talk competition winners. So in third place, winning a prize of $100, we have Lushan. Good job, Lushan. It was definitely well earned. Then coming up in second place, winning a total of $150, we have Gerline. Congratulations, Gerline. You did a really great job. And then finally, in first place, rather, winning the grand total of $200, we have Julian. So congrats, Julian. Way to go. You really gave an awesome presentation, and I think everybody agrees with that. So, so after this symposium, we'll go ahead and we'll be getting in touch with you three winners so that we can really figure out the logistics of getting that prize money out to you guys. So, yep, let's everybody take one more quick moment to give our winners a virtual round of applause. Great job, you guys. And to all our competitors today, thank you guys so much for your participation. Even though you may not have won, you all really did a great job. I think we all really learned a lot. And so with that, I'll go ahead and I'll send things over back to Josh to wrap things up. Thank you, everybody.
Thank you, Jordan. I would like to begin these closing remarks once again by thanking all of our invited speakers and agreeing for taking the time out of their busy schedule to share their knowledge and experience with us. I would also like to thank all the members of our Plant Science Council here at the University of Florida. Many hands contributed to everything that made the symposium a success. And I would also like to thank all of you who registered and participated in this year's symposium. Can't go without thanking Corteva for giving us this great opportunity and helping us put it together. They help forge these connections that help grow our knowledge as scientists, and it will really affect our careers and our future. Over these last 2 days, we learned about plant stress and its impact on crops and people, how important it is where plants grow to our culture, and the importance this plays with native nations, the amazing work being done to unravel and model complex genetic structures affecting plant resilience, the important role plant breeding has in the world stage for feeding the future, and the need for us as scientists to advocate for sound, value-driven technologies. Plant biology continues to amaze us. The work being done to understand critical plant phenology is incredible. We have learned from fellow graduate students as they showed us some of their sweet communication skills and showcased their research and experience for us. And as we are all grateful for this very well hammered out case study of a solid hypothesis test. Science. Isn't it cool? Was it not great to be so motivated by that last talk? I have thoroughly enjoyed this year's event and hope to see you again at the 7th annual Plant Science Symposium next year. I think we might even need to do a little name change. We will conclude this session now But again, check in with us again at 2:30 Eastern time on our other Zoom link to participate in a workshop to glean some knowledge from experienced experts on budgeting and managing resources in professional settings. Thank you for your participation. And unless I can ask Jason if he wants to say any last words.
Nope, no last words. It was a fantastic symposium. Enjoyed the last 2 days and some great speakers. So congratulations to all the students and all the organizers. It was a great couple of days. So I just hope we can do this in the warm Florida weather next year.
Yeah, I completely agree. So thank you again and have a great rest of your